Hello everyone, welcome back to IMAT-Tutor. Today we will discuss lecture 11 where we are diving into the fascinating world of digestion and absorption. Essentially, we're looking at the whole journey from the first bite to the bloodstream by understanding how the body dismantles food and captures energy. It's an incredible process, and today we have quite a few subtopics covered on our agenda. First, we will take a look at the overview and organization of the alimentary canal to see how everything is laid out. Then we will start right at the top with the mouth, focusing on mechanical digestion and salivary amylase. Moving down, we will explore the esophagus, specifically looking at peristalsis and the lower esophageal sphincter. Next up is the stomach, where we will break down acid, pepsin, and gastric motility. After that, we will shift our focus to the helper organs. We will discuss the liver and bile, including emulsification and bile salt recycling. Then we will examine the pancreas, comparing its endocrine versus exocrine function and digestive enzymes. That brings us to small intestine, where we will study its structure, brush border enzymes, and final digestion. While we are there, we will unpack the precise absorption mechanisms, including active transport, facilitated diffusion, and micelles. Next, we will move to the large intestine to cover water absorption, microbiome, and defecation. To understand how this all is coordinated, we will look at the hormonal control, specifically gastrin, secretin, cholecystokinin, as well as nervous control, featuring the enteric nervous system and vagus nerve. Finally, to wrap it all up, we will tackle 10 IMAT-style MCQs with full explanations and review everything with a master summary table. So let's get started. If you want IMAT biology notes that actually help you score, that are not bulky textbooks, not random summaries, but exam-focused, trap-oriented, high-yield PDFs, the link is in the description. Study smart, not hard. Let me start by giving you the big picture before we zoom into any single organ, because the biggest mistake students make with digestion is learning it as a disconnected list of enzymes. That approach will fail you on the IMAT. Instead, to understand the human digestive system as depicted on these figures, we build our concept from first principles by mapping out a continuous specialized muscular tube known as the alimentary canal. We position the start of this canal at the mouth and follow its path until it terminates at the anus, spanning an approximate total length of 9 meters. Along this pathway, we place accessory organs, specifically the salivary glands, liver, and pancreas, which do not directly handle food transit, but instead secrete vital chemical inputs into the lumen to facilitate breakdown. This is the main pathway of food transit. As the intestine is twisted, so we are just roughly showing the pathway inside the intestine. Then it comes to large intestine and it travels like this up until it reaches anus. Now let us build the microscopic architecture of this tube wall from the inside out, which is seen on the figure 11.2, where 4 distinct concentric layers are established. Directly facing the internal lumen, we locate the mucosae, which is a composite barrier comprising a surface epithelium. This is the surface epithelium of mucosae. Then we have an underlying supportive connective tissue layer called the lamina propria, indicated by our pointer. This whole region is lamina propria. You will also see some localized patches of defensive lymphatic tissue, the green T-shaped structures. These are the lymphatic tissues. You will also see deep gland, one over here, another here, and finally a thin smooth muscle layer termed muscularis mucosae. Moving outward, we position the submucosae, a dense layer of connective tissue housing a distinct vein. This is the vein we're talking about, although it has been cut off going forward, and also artery and nerve network, complex glands, one over here, another here, and the intricate network of submucosal plexus called the plexus of Meissner, which regulates local secretions. External to the submucosae, we establish muscularis, structured as an inner circular layer and an outer longitudinal muscle layer. Nested directly between these two muscle sheaths, we find the myenteric plexus, which serves as the primary motor driver for coordinating functional contractions. Finally, wrapping the entire structure on its outermost surface, we add the serosa, a protective layer composed of areolar connective tissue and a surface epithelium, supported by anchoring mesentery, which occasionally interacts with a duct of a gland outside the tract. Now let us map out the structural sequence from top to bottom. At the entrance, we already have located the mouth, containing the palate, uvula, tongue, and teeth, flanked by 3 pairs of salivary glands, i.e. sublingual gland, submandibular gland, and parotid gland. They are in total 6 in number. These elements cooperate for mechanical breakdown and initial starch digestion using salivary, amylase, and lubricating mucin. Below the mouth, the canal transitions into the pharynx. This part is the pharynx we're talking about. The pharynx then transitions into the esophagus, which starts just over here and continues up to the start of the stomach. This is a region dedicated purely to transport via wave-like peristalsis, and this esophagus is guarded at its distal end by the lower esophageal sphincter, or LOS. We will talk about the sphincter later in detail. The esophagus then expands into the stomach, an environment optimized for protein denaturation and primary cleavage. This chamber utilizes hydrochloric acid to establish a low pH, activating the Zymogen Pepsinogen into the active protease pepsin, while simultaneously releasing intrinsic factor, or IF. Just below the stomach, we enter 3 sections of the small intestine. First, we position the C-shaped duodenum. This is the duodenum we're talking, where aggressive neutralization of gastric acid takes place alongside enzyme digestion, heavily supplied by pancreatic duct, delivering pancreatic juice, and common bile duct, delivering bile from the liver and gallbladder. Moving forward, we find the jejunum, which is a long twisted tube, roughly starting over here, and it continues a long way before it transitions to become ileum over here. Jejunum is specialized for absolute majority of nutrient absorption due to its extensive brush border enzymes. And as we were saying, we conclude the small intestine at the ileum, which is structurally adapted for the specialized absorption of vitamin B12 and the critical recycling of bile salts. Finally, the remaining material is delivered into the large intestine, which we segment into the cecum at first, which anchors the small blind-ended appendix, followed by the ascending colon. This part is the ascending colon. Then comes transverse colon and descending colon, sigmoid colon, this part, and the final part is the rectum, which stores waste prior to elimination through the anus. This final region lacks internal villi entirely and contains abundant goblet cells, specialized for water recovery and supporting a massive symbiotic microbiome. Having established this complete conceptual blueprint, we will now transition directly to the precise terms, structural parameters, and high-yield criteria displayed on the anchor slide 1. As established in our structural layout, the alimentary canal is defined as a continuous muscular tube extending from the mouth to anus with an average biological length of approximately 9 meters. Functioning alongside the main pathway, the accessory organs, consisting of the salivary glands, liver, and pancreas, perform their digestive roles by discharging specialized chemical secretions directly into the canal lumen. The gastrointestinal tract maintains a universal 4-layered architecture organized from the lumen outward as mucosae, submucosae, muscularis externa, and serosa. The innermost layer, or mucosae, faces the open lumen directly and contains an internal epithelium, an underlying connective tissue layer known as the lamina propria, localized patches of defensive lymphatic tissue, and a thin smooth muscle layer called muscularis mucosae alongside deep structures like a gland in the mucosae. Positioned immediately beneath the mucosae, the submucosae layer serves as a supportive connective tissue highway, housing a distinct vein, artery, and nerve network, complex glands in the submucosae, and the highly specialized submucosal plexus, also known as the plexus of Meissner, which acts as the localized regulator of the glandular secretions. The primary contractile engine, or muscularis layer, is split into an inner circular muscle layer and an outer longitudinal muscle layer, which house the centrally situated Auerbach's plexus or mesenteric plexus, responsible for driving gut motility while receiving a supportive connection from the anchoring mesentery. For information, mesentery is a structure that holds abdominal structures in place, or it works like a hammock for your intestines. The fourth layer, called serosa, encapsulates the external surface of the tract, and the serosa layer provides structural integrity and reduction of friction through a dual component layer consisting of areolar connective tissue and a surface epithelium, occasionally interacting with a duct of gland outside the tract. Now, the entire digestive tract or alimentary canal is divided into specialized zones with unique features, starting with the mouth, which incorporates the protective palate, uvula, muscular tongue, and sharp teeth, cooperates with the sublingual, submandibular, and parotid salivary glands to manage mechanical breakdown and starch digestion using salivary, amylase, and mucin. The esophagus provides transport only via peristalsis and the lower esophageal sphincter. The stomach orchestrates protein denaturation and pepsin activation via hydrochloric acid, pepsinogen, and intrinsic factor. The duodenum drives neutralization and enzyme digestion via pancreatic juice and bile. The jejunum executes the majority of nutrient absorption using brush border enzymes. The ileum specializes in vitamin B12 and bile salt reabsorption. That's why in key feature it is written terminal ileum is specialized for this B12 and bile absorption. Finally, the large intestine coordinates water recovery and microbiome maintenance utilizing a villi-free mucosae rich in goblet cells. Pay critical attention to these high-yield exam traps to protect your score. Examiners love to trick students regarding the absorptive capacity of the stomach. You must remember the trap that stomach absorbs almost nothing nutritionally, with the strict physiological exceptions of alcohol and aspirin. Speaking of aspirin, which is a salicylate class drug, this is used for pain relief and antiplatelet therapy. Watch out for questions that swap the anatomical locations or alternative names of the intrinsic nerve plexuses. Ensure you link Meissner's plexus exclusively to submucosa and map the mesenteric plexus or Auerbach's plexus strictly between the circular and longitudinal layers of the muscularis externa. A favorite exam question involves the mucosal surface of the colon. Examiners often falsely describe the large intestine as containing villi to increase surface area for water absorption. Do not fall for this. The large intestine has no villi and relies heavily on goblet cell for lubrication. And finally, the take-home exam pearls. The first one talks about the second brain. Both the submucosal and mesenteric plexuses together constitute the enteric nervous system, or ENS. It is clinically celebrated as the second brain because it possesses autonomous reflex arcs capable of managing gastrointestinal secretion and motility, completely independent of central nervous system input. Pepsinogen is released as an inactive zymogen to prevent the auto-digestion of the gastric or stomach wall, requiring highly acidic environment created by hydrochloric acid secretion to undergo structural cleavage into functional pepsin. Vitamin B12 absorption requires binding to gastric intrinsic factor or IF in the stomach, but the physical uptake of this complex is entirely restricted to specialized receptor pathways located in the terminal ileum. Now we will examine the oral physiology shown in the following images, and it involves establishing the fundamental architecture of mastication and evaluating how chewing drives initial digestion. This approach builds a conceptual understanding of the system's structural mechanics. We locate the structural breakdown within the mouth, where specialized tools for fragmentation initiate mechanical processing. Here we position the incisors to cut, the canines to tear, and molars to grind food into smaller particles. You can look at this following shortcut to memorize the number of teeth and their type. This shortcut shows the standard dental formula for permanent human teeth, organized into 4 quadrants, 1, 2, 3, and 4. Each quadrant contains 2 incisors, 1 canine, 2 premolars, and 3 molars. Adding this together gives 8 teeth per quadrant, totaling 32 adult teeth in the entire mouth. Simultaneously, we add the roll of the tongue, which shapes food into moistened mass to form a bolus and prepare it for swallowing. And this mechanical division creates a massive biochemical impact. By breaking large food chunks into smaller pieces, we are maximizing surface area, which exposes more substrate molecules for enzymes to attack simultaneously. This surface exposure directly causes an enhanced catalytic efficiency, allowing for faster enzymatic digestion per unit of volume. Next, we introduce the fluid environment of the oral cavity by mapping out the salivary glands. We establish 3 distinct pairs: the parotid, which is the largest, the submandibular, and the sublingual glands. Together, these glands produce a volume of 1 to 1.5 liters of saliva. Saliva is composed primarily of water, but we must also place several crucial biochemical components into this mix. We include mucin, which acts as a glycoprotein lubricant to ease bolus passage; bicarbonate, which buffers oral acid for dental hygiene and maintains an optimal oral pH of approximately 6.8 to 7, and lysosome alongside immunoglobulin A, which provide essential antimicrobial oral defense. Finally, we add lingual lipase and salivary amylase, which is the star of oral chemical digestion. And this salivary amylase was historically known as ptyalin. Let us map out the enzymatic mechanism and path of salivary starch digestion. For your information, starch is a polysaccharide composed of amylose and amylopectin. When salivary amylase encounters starch, it displays strict bond specificity, meaning it exclusively breaks internal bonds. This process is designated as alpha-1,4-glycosidic cleavage, which breaks down long chains to produce shorter segments called dextrins and maltose. However, we hit a major structural limitation when dealing with amylopectin branching. Starch contains alpha-1,6 bonds that remain completely untouched by salivary or pancreatic amylase because the enzyme only cleaves alpha-1,4 bonds. This leaves branch points intact. 1,6 branch points are left intact as they move down the tract. That means the amylopectin or alpha-1,6 branch points will not be digested or cleaved in oral cavity or stomach. It will finally get cleaved or digested when it approaches small intestine. Following the path of the bolus past the oral cavity, we observe stomach inactivation. The stomach contains gastric acid that drops the environmental pH down to a highly acidic range of 1.5 to 3.5, or more broadly less than 3. Because salivary amylase requires a near neutral environment to function, digestion pauses at this stage and only resumes later in the duodenum. The uncleaved alpha-1,6 branch points must travel all the way to the small intestine, where an alpha-dextrinase requirement is established, meaning these specific branch points are only cleaved by enzymes like isomaltase located on the intestinal brush border. Having mapped out this entire physiological journey from the mouth to the initial stages of gastric transit, we will now transition directly to the precise definitions, key sequence values, and high-yield criteria displayed on the anchor slide 2. As established during our conceptual layout, salivary amylase or ptyalin is specifically defined as an alpha amylase, which is responsible for cleaving internal alpha-1,4 glycosidic bonds. You can remember easily that the name amylase or amylase breaks amylose of starch. Then the slide defines the bolus as a completely moistened chewed food mass that is formed by the tongue and is ready for swallowing. The salivary gland infrastructure consists of 3 main pairs, with the parotid being the largest, followed sequentially by the submandibular and sublingual glands. Talking about saliva, the physiological normal output ranges from 1 to 1.5 liter per day, consisting of water, mucin, amylase, lingual lipase, lysozyme, immunoglobulin A, and bicarbonate. And the core feature of mastication is that it mechanically increases total surface area to maximize direct enzyme contact. The biochemical pathway of starch digestion follows the sequence where starch comprising amylose and amylopectin reacts with amylase to yield dextrins and maltose. The amylase we're talking about only cleaves the alpha-1,4 bonds inside the starch. To be more precise, it breaks the amylose only. The alpha-1,6 branch points are not cleaved by amylase, rather they are handled by isomaltase, which is located in the brush border of enterocytes. More on that later. And the operational sequence requires an optimal pH of 6.8 to 7, and the enzyme is completely inactivated once it encounters a gastric acid pH which is less than 3. Now we will move to the high-yield exam traps, and you need to master these critical testing pitfalls to secure your score. Examiners frequently try to trick students by stating that salivary amylase can fully digest all components of starch. You must remember the trap that amylase cleaves alpha-1,4 bonds only. It cannot break alpha-1,6 branch points, and those specific amylopectin branch points are left completely untouched until they encounter isomaltase in the intestinal brush border. Watch out for questions that claim amylase is inactivated in the duodenum due to pancreatic secretions. The slide explicitly marks the trap that amylase is inactivated early in the stomach due to low gastric pH, not the duodenum. And do not confuse the function of mucin with bicarbonate. Mucin functions strictly as a glycoprotein lubricant for mechanical transit, whereas bicarbonate acts as a chemical buffer to manage oral acid and preserve dental hygiene. And to master the high-yield physiological mechanisms tested on the IMAT, let's analyze these core medical breakdown principles. The first pearl talks about the activity of amylase. Although gastric acid inactivates ptyalin, a brief window of amylase activity actually continues inside the stomach because the dense core of the swallowed bolus temporarily shields the internal enzymes from acid penetration. Then we will talk about the catalytic efficiency rule. The fragmentation of food by teeth is not just a mechanical convenience. It is a mathematical prerequisite for digestion because increasing the surface area to volume ratio exponentially raises the number of parallel enzymatic collisions per unit of time. And to understand the final exam pearl, we will look at this diagram. This whole diagram tracks the layers of small intestine from a macrostructural outline of the tissue folds called villi to the individual epithelial cells called enterocytes. Imagine this is a segment of intestine and it has villi in it. Villi is plural for villus. We take one villus and we zoom it in to see what's inside. You will see enterocytes and you will also see highly congested hair-like outer edge of the cells, which represents a brush border membrane or also known as microvilli. This microvilli is a structural adaptation engineered to massively amplify surface area for terminal chemical digestion and nutrient absorption. Complex branched carbohydrates like glycogen and amylopectin component of dietary starch contain two primary types of linkage. One is continuous linear alpha 1,4 bonds and branch point, which are alpha 1,6 bonds. If we draw it here, let's say this is glucose, these bonds are alpha 1,4, but these bonds are alpha 1,6 branch points. Soluble digestive enzymes secreted into the gut lumen like pancreatic alpha amylase or the salivary amylase are highly specialized. They can easily break the linear alpha 1,4 bonds, but they cannot hydrolyze alpha 1,6 branch points. Because alpha amylase bypasses these branch points, it leaves behind tangled, partially digested carbohydrate fragments known as limit dextrins. Complete digestion relies completely on the brush border for the following reasons. The first reason is location of the debranching enzymes. The specific enzyme required to tear down this branch point is isomaltase that we have mentioned already quite a few times, also referred to as alpha dextrinase. As the limit dextrins brush against the enterocytes, the active site of the embedded isomaltase physically locks onto and hydrolyzes the structural alpha 1,6 glycosidic links. These red hexagonal shapes represent the alpha 1,6 links, and isomaltase breaks this specific alpha 1,6 bond, and breaking these branch points yields free glucose monosaccharides right at the membrane surface. This allows immediate transport across the enterocyte wall into the bloodstream before the free sugars can be consumed by intestinal bacteria. And that's why we say amylopectin has brush border reliance or complete breakdown. Now let's transition into how food actually moves through the upper GI tract, focusing on peristalsis and the lower esophageal sphincter. We will analyze the upper GI tract from scratch to map out how the esophagus works and transports food. We introduce the esophagus as a specialized muscular passage, measuring approximately 25 centimeters in length, which functions purely as a transport zone, executing no baseline chemical digestion or nutrient absorption. On the microscopic layout, we establish its protective surface barrier by placing a stratified squamous epithelium. The structural design forms a tough, abrasion-resistant lining specifically optimized to withstand ongoing mechanical wear and tear from passing food boluses. In this image, you see the food bolus passing through esophagus, and it's getting protected by the structure of stratified squamous epithelium, which is abrasion resistant. This epithelial structure is different from the structure of the stomach, which has simple columnar epithelium. At the most distal end of this conduit, we position the lower esophageal sphincter. This is the LOS, or lower esophageal sphincter, which is also clinically referred to as the cardiac sphincter. Under resting conditions, we map this muscular zone as totally contracted, creating a tight physical barrier that prevents the reflux of highly corrosive gastric acid into the upper tract. In this image, you see this is the stomach acid here, and it's getting prevented by the lower esophageal sphincter due to its contraction. Let's map out precise timing and movement patterns that dictate how food enters and travels through this passage. Breaking down the 3 phases of deglutition. The event begins with the oral phase, which relies on voluntary tongue propulsion. This is the tongue, which does the action of propulsion to push the food bolus backward. As the bolus hits the back of the mouth, roughly here, it triggers the pharyngeal phase, which is an involuntary reflex sequence requiring highly coordinated structural movements to protect the respiratory pathway. During this pharyngeal stage, we locate 2 crucial protective actions. The soft palate, this is the soft palate, and this palate rises to completely close off the nasopharynx, preventing food from entering the nasal cavity, while simultaneously the epiglottis, this is the epiglottis we're talking about, and this epiglottis depresses to cover the larynx, fully sealing the airway. Once the bolus clears the pharynx, we activate the esophageal phase, and the esophageal phase will be seen throughout the esophagus, and this is entirely involuntary. This transport stage is powered by esophageal peristalsis, a highly organized wave of smooth muscle coordination orchestrated directly by Auerbach's plexus. To visualize the precise mechanics of propulsion and protection, we can establish a dual action wave moving along the tube. We place a ring of coordinated circular muscle contractions directly behind the food bolus, that means here, which will push the food bolus forward, while simultaneously establishing a zone of relaxation ahead of the bolus here, complemented by longitudinal muscles that contract to shorten the path length. As this involuntary peristaltic wave arrives at the base of the conduit, that means around here, it forces the tonically contracted lower esophageal sphincter, this sphincter, to relax. This action opens the gateway for the bolus to enter the gastric chamber. That means it comes inside the stomach. And after this, the sphincter promptly contracts again to block stomach acid. This mechanical system reveals a fundamental physics principle. Peristalsis is gravity independent because the propagation relies on an active localized muscular process rather than passive downward falling. This transport remains fully operational in microgravity or even if an individual is positioned upside down. If this protective mechanism fails, we observe the development of GORD or GERD, known as gastroesophageal reflux disease, which is a structural failure or loss of baseline LOS tone that allows highly acidic gastric contents to flow backward and damage the unprotected, vulnerable esophageal lining, which lacks intrinsic acid defenses. That means the stratified squamous epithelial cells are assaulted by the gastric acid. We have completely mapped out this conceptual framework from the pharynx down to the gastric junction. Now we will transition directly to our anchor slide. The slide defines the esophagus as an approximately 25 cm conduit whose primary physiological profile involves performing no digestion or absorption. Now coming to lower esophageal sphincter, which is also known as cardiac sphincter, the muscle group remains tonically contracted under basal conditions to actively prevent reflux. When it is said that tonically contracted, that means that it is always contracted in a baseline tone. Now coming to the epithelial specialization. The histological layout features an abrasion-resistant stratified squamous epithelium, a protective design that is completely unlike the simple columnar epithelium characterizing the stomach wall. The propagation of regular propulsive waves throughout this esophagus is directly coordinated by the intrinsic network of Auerbach's plexus or mesenteric plexus. And the physiological mechanism of swallowing moves sequentially through three distinct phases: a voluntary oral phase, followed by an involuntary reflex pharyngeal phase, terminating in an involuntary esophageal phase. And during the pharyngeal sequence, airway protection is achieved because the epiglottis covers the larynx, while the soft palate simultaneously closes off the nasopharynx. And now we will talk about the peristaltic motor rule. This functional sequence requires a coordinated circular muscle contraction behind the bolus paired with simultaneously relaxing action ahead of it. And the lower esophageal sphincter responds dynamically by relaxing as the peristaltic valve arrives, opening to permit bolus entry and immediately contracting again. Now let me present the high-yield exam traps, and if they are learned right, they will protect your exam score. The first trap talks about the gravity dependence misconception. Examiners routinely attempt to confuse students by stating that esophageal transit relies heavily on gravity or posture. You must remember the trap that peristalsis does not require gravity because it is an active muscular process. It functions perfectly in microgravity or when upside down. Then watch out for questions that mischaracterize the cause of acid-induced chest pain. The trap focuses on LOS failure leading to GORD because the stomach acid escapes onto a stratified squamous epithelium that possesses absolutely no intrinsic acid protection. It results in severe tissue irritation and burning pain. Examiners love to swap the locations of epithelial tissue type as well. Ensure you never confuse the stratified squamous epithelium, which is the lining of esophagus, with the simple columnar epithelial lining that initiates abruptly at the gastroesophageal junction. The esophageal epithelium stands out as highly unique because it is stratified squamous, making it completely non-glandular and a distinct structural exception within the primary segments of the GI or gastrointestinal tract. While Meissner's plexus handles mucosal secretions, the coordination of the muscularis external layers during a swallow relies entirely on Auerbach's plexus to generate the highly organized circular and longitudinal contractions, and the temporary elevation of soft palate and depression of the epiglottis during deglutition are mandatory hardwired neural reflexes, ensuring that nutrient transport completely overrides the respiratory pathway for a fraction of a second to eliminate aspiration risk. We already have discussed in the respiratory system lecture that aspiration means a food bolus or foreign particle getting inside the respiratory tract, which can be life-threatening or fatal. And this problem is solved by the reflexive protection priorities maintained by soft palate and epiglottis. Now we will move our attention to stomach, which is the most chemically aggressive organ in the digestive system, and it earns that reputation by maintaining a luminal pH of around 1.5 to 3.5. That is more acidic than battery acid for comparison. To understand the physiological framework of gastric system, we build our concept by analyzing the stomach's anatomy and secretory physiology. We begin by positioning the macroanatomical region of the stomach, establishing the upper fundus, the central body, and the lower antrum. We locate the body as the primary secretion hub, which serves as the major site for producing gastric juices. Below the body, we place the antrum, identifying it as the motility center responsible for coordinating stomach movement and driving gastrin release. Gastrin is a stomach hormone. Looking closely at the mucosal surface, if we take a cross-section of the stomach and we look at the mucosae, we introduce gastric pits, which are specialized mucosal invaginations lined with specialized secretory cells that descend into the tissue wall. Within these functional units, we locate and position 5 highly specialized cell types, each contributing a unique biochemical input. First, we add the mucus cells, orange or yellow cells near at the top neck of the pit, which produce mucus to protect the stomach lining from acidic damage. Next, we establish the parietal cells, also known as oxyntic cell, which secrete both hydrochloric acid and intrinsic factor. This intrinsic factor is a molecule required for downstream vitamin B12 absorption. Below this, we place the chief cells, also known as zymogenic cells, the light blue cells at the very bottom of the pit, which secrete pepsinogen, the inactive precursor to the protein digestive enzyme pepsin. We also add ECL cells or enterochromaffin-like cells. Though they are not explicitly labeled in our diagram, you must know that ECL cells secrete histamine to act as a localized paracrine parietal cell stimulant. Finally, we position G cells, the green one located near the lower section of the pit, which secrete hormone gastrin into the local circulation. Let us map out the precise cellular mechanism of hydrochloric acid production occurring within the parietal cell. The sequence initiates as carbon dioxide and water combine inside the cytoplasm to form carbonic acid, catalyzed by the enzyme carbonic anhydrase. This carbonic acid rapidly dissociates into hydrogen ions and bicarbonate ion. The resulting proton is actively pumped out of the cell into the gastric lumen by an ATP-driven transport protein, which is called the proton potassium ATPase pump, and chloride ions or Cl- follow through separate channels into the lumen to form functional hydrochloric acid. Simultaneously, the newly generated bicarbonate ion HCO3- exits the basal membrane of the cell and enters the blood vessel or bloodstream after a meal, creating a phenomenon known as the alkaline tide, which causes a transient blood pH rise. This production of HCl serves multiple critical functions. It denatures protein structures to expose their peptide bonds, and it actively cleaves and activates pepsinogen into functional pepsin. It kills ingested bacteria and establishes the optimal pepsin pH for enzymatic cleavage. As these chemical processes occur, the vigorous mechanical movement of the muscular walls mixes the food, converting it into a semi-liquid paste produced by gastric churning called chyme. Having mapped out these physiological pathways and chemical mechanisms, we will now transition directly to the explicit text definitions and core structural values listed on our anchor slide. As we pointed in our structural layout, gastric pits are defined as specialized mucosal invaginations lined with specialized secretory cells. Now that we see where these secretions happen, let's look at what they mix with, specifically chyme. The slide defines chyme as the characteristic semi-liquid paste produced by active gastric churning. Now we talk about 5 types of cells found on the gastric mucosal layer, and we will talk about them under the headlines of cell type, secretion, and function. First comes parietal cell, which is also designated as oxyntic cells, and these elements secrete hydrochloric acid and intrinsic factor to execute protein denaturation and enable vitamin B12 absorption. Alongside acid production, the stomach also needs enzymes, which brings us to chief cells, alternately named zymogenic cells. The primary role is the secretion of pepsinogen, which functions as the direct precursor to active pepsin. To control and trigger these various secretions, the stomach uses regulatory units called G cells. These specialized cells secrete the hormone gastrin, which travels humorally to stimulate gastric hydrochloric acid secretion and overall motility. With all this acid and enzyme activity, the stomach must protect itself, which is where mucous neck cells come in. Positioned within the pits, these cells secrete mucus that actively protects the stomach lining and mucosa from acidic damage. Beyond mucus protection, the stomach fine-tunes its acid levels locally using ECL cells. The secretory units produce histamine, which operates locally as a paracrine parietal cell stimulant to drive acid production. The biochemical pathway of hydrochloric acid production relies on this following sequence. First, carbon dioxide reacts with water to make carbonic acid with the help of carbonic anhydrase enzyme, supplying H+ or proton to be pumped into the lumen by proton potassium ATPase alongside chloride ion. As a direct byproduct of pumping this acid into the stomach, we see a temporary shift in blood pH known as alkaline tide. The physiological movement of bicarbonate into the blood vessel after a meal creates a transient blood pH rise termed the alkaline tide. And as we have talked about, HCl functions are denaturation of protein, activation of pepsinogen to pepsin, killing of bacteria, and setting up optimal pH for pepsin activity. Master these critical exam traps to safeguard your hard-earned points. When talking about the first trap, you gotta know examiners frequently attempt to link intrinsic factor secretion to cheap cells because of its role in enzyme adjacent nutrient processing. You must remember the trap that intrinsic factor is produced exclusively by parietal cells, meaning any disruption to the axanthic lineage halts both acid production and vitamin uptake pathways simultaneously. And watch out for the questions describing clinical parietal cell destruction or autoimmune targeting. This pathology never causes an isolated deficit. It always manifests concurrently as both achlorhydria and pernicious anemia caused by the absolute loss of intrinsic factor mediated absorption. Now that we understand these clinical conditions, let's pivot to some high-yield exam pearls you are guaranteed to see tested. While the standard segment of the alimentary canal utilizes only 2 layers of smooth muscle, the stomach is uniquely adapted with 3 complete muscle layers, the oblique, circular, and longitudinal layers, providing the specialized mechanical power required for vigorous churning and chyme formation. And also, hydrochloric acid does not digest proteins independently. Its primary clinical value lies in dropping the luminal pH. That means inside the stomach, hydrochloric acid drops the pH, which creates the strict environment required to activate pepsinogen and maintain the narrow optimal pH window that pepsin demands for catalytic cleavage. Now we shift our gear towards pepsin and gastric motility. Pepsin is the stomach's protease. That means pepsin is responsible for breaking down protein inside stomach. But I want you to understand from the outset that pepsin is the opening act, not the headline performer of protein digestion. Students overcredit pepsin because it is the most famous gastric enzyme. But if I asked you to rank the contribution of all proteases to complete protein digestion, pepsin would come third, behind pancreatic trypsin and chymotrypsin. Pepsin starts the job, makes the first cuts, and then is destroyed in the duodenum before it can finish anything. Keep that hierarchy in your head. And so, to understand the physiology of gastric digestion, its enzymes, and its clinical pitfalls, we built our concept by establishing the chemical activation engine of the stomach. We position a parietal cell powerhouse next to a chief cell within the gastric mucosa. The parietal cell actively pumps hydrogen ions into the lumen. When parietal cell pumps hydrogen ions into the lumen, the pH drops to the range of 1.5 to 2.5, or broadly a pH less than 2. Simultaneously, the chief cell secretes pepsinogen, which is an inactive Zymogen. When pepsinogen encounters the low luminal pH created by hydrochloric acid, it undergoes a conformational shift that cleaves off its protective masking sequence, transforming it into active enzyme pepsin. Once initial pepsin molecules are formed, they interact directly with remaining pepsinogen molecules to accelerate this conversion via autocatalytic pepsin activation. That means pepsin does the role of activating other pepsinogens. So we can write pepsin reacts with pepsinogen and it creates more pepsin like this. As the parietal cell generates acid, it yields an internal byproduct, which is bicarbonate or HCO3-. We locate the transport pathways that move this bicarbonate out of the basal side of the cell and into the adjacent blood vessels. That means from the cells, they come to the adjacent blood vessels, which triggers a localized blood pH rise known as the alkaline tide. Biochemically, pepsin operates specifically as an active endopeptidase, meaning it cleaves internal peptide bonds rather than terminal ones. We position targeting specificity of pepsin at bonds associated with aromatic or hydrophobic residues, specifically targeting phenylalanine, tryptophan, and tyrosine bonds. Because it cleaves internally at the selective sites, the resulting products are exclusively polypeptide fragments, not free amino acids. And based on that information, we map out the clinical realities and misconceptions surrounding the protein digestion trap. While many believe the stomach is the primary site of protein breakdown, pepsin only handles the initial digestion. We contrast this with the pancreatic enzymes, such as trypsin or chymotrypsin, which perform the dominant breakdown in the small intestine to finally yield free amino acids. As the chyme moves forward, we locate pyloric sphincter, a muscular gateway that controls the rate of chyme release into the duodenum. When this material enters the duodenum, it encounters an alkaline pH of around 6 to 8. This rapid shift in environmental condition causes the pepsin enzyme to undergo irreversible denaturation, which permanently deactivates or inactivates pepsin. To regulate this transit, we establish the enterogastric reflex, a neural and hormonal feedback loop triggered when fat, acid, or hypertonic chyme enters the duodenum, which acts to slow gastric emptying. Furthermore, we locate the pathways of selective gastric absorption that is seen in this segment. While general nutrients are not absorbed here, the low pH keeps specific lipid-soluble compounds non-ionized, allowing for the direct absorption of lipid-soluble ethanol and aspirin. Finally, we establish the pernicious anemia connection, where autoimmune destruction of parietal cells leads to severe acid loss and a lack of intrinsic factor, causing a downstream vitamin B12 deficiency. With the mechanical and biochemical framework established, let's dive into the core terms and high-yield criteria on our anchor slide. The slide starts its introduction with pepsinogen, and it defines pepsinogen as an inactive Zymogen secreted by chief cells that requires HCl at a pH less than 2 to undergo initial cleavage into its active form. Once activated, how do we classify this powerful enzyme? And to get that answer, we come to pepsin. Functioning as an active endopeptidase, pepsin is biochemically specialized to cleave internal peptide bonds at aromatic or hydrophobic residues. Let's look exactly at the amino acid bonds this endopeptidase breaks. The enzymatic mechanism relies on a sequence where pepsin targets phenylalanine, tryptophan, and tyrosine bonds. What specific molecules result from this targeted sequence cleavage? The chemical action yields polypeptide fragments as products, explicitly noting that these do not represent free amino acids. The fragments then move forward, but what happens when pepsin hits the duodenum? Upon entering the alkaline pH of the duodenum, which ranges from 6 to 8, pepsin undergoes immediate and irreversible denaturation. The stomach controls this entry into the duodenum using a specialized muscular boundary known as pyloric sphincter that sits at the end of the stomach boundary. And this muscular boundary serves as the primary anatomical mechanism that actively controls the rate of chyme release into duodenum. And this boundary works closely with neural feedback to optimize chyme release. And the neural feedback we can address as enterogastric reflex. This reflex is a physiological sequence initiated by the presence of fat, acid, or hypertonic chyme in the duodenum that slows overall gastric emptying to optimize digestion. Now let's shift to the tricky traps examiners use to test this reflex and digestion process. Examiners frequently try to catch students off guard by asserting that pepsin is the main protease responsible for complete protein degradation. You must remember the trap that pancreatic trypsin and chymotrypsin do the heavy work of dominant breakdown, while pepsin only handles initial processing. Beyond who does the heavy lifting, examiners also love to trick you on the exact end products. Watch out for questions stating that gastric digestion yields free amino acids. The slide explicitly highlights that pepsin produces peptide fragments, not free amino acids. Don't get confused about this. While we only have fragments here, let's clarify what the stomach actually can absorb. Do not fall for the trick that the stomach is a major nutrient absorptive organ. The text reinforces that standard nutrients are absolutely not absorbed here, with the selective exceptions of lipid-soluble ethanol and aspirin. Let's conclude by solidifying the absolute highest yield pearls you must memorize for the exam day. The first one would be the autocatalytic trigger. The conversion of pepsinogen is highly efficient because it is autocatalytic once started. The presence of newly formed active pepsin works in a positive feedback loop to cleave remaining zymogens or pepsinogens to make more pepsin. Then comes the pernicious anemia. When this acid-driven system fails entirely due to autoimmune destruction, we see classical clinical triad. Autoimmune destruction of the parietal cell powerhouse creates a devastating clinical triad consisting of direct acid loss, a complete block in intrinsic factor reabsorption, and a resulting macrocytic vitamin B12 deficiency, or we can say pernicious anemia. While a lack of acid ruins vitamin B12 absorption, a low pH actively drives the absorption of specific weak acids. Aspirin and alcohol that we have already said are absorbed directly across the gastric epithelium because the low luminal pH maintains them in a protonated lipid-soluble state, allowing them to rapidly cross the plasma membrane of the enterocyte. If you master these distinct physiological triggers and pathways, you will easily crush any GI question on exam day. Before we enter the small intestine, we need to understand two accessory organs that pour their secretions into the duodenum, and they are liver and pancreas. To evaluate the physiology of lipid processing, we will start by analyzing bile as the multitasking mechanic of fat digestion. We introduce bile not as a chemical enzyme, but as an amphipathic detergent secretions from the liver. That means don't get confused with the production site of bile. It is liver who produces bile, and the bile is stored in gallbladder. The production rate of bile tracks at approximately 600 to 1000 milliliter per day, and it is sequentially stored and concentrated within the gallbladder that we already mentioned. Let us map out the chemical composition of this bile. It is composed of water, bicarbonate, phospholipid, cholesterol, the breakdown pigment of bilirubin, and bile salts. We position these bile salts as cholesterol derivatives. That means these bile salts are produced from cholesterol, and some common examples or major forms are sodium taurocholate and sodium glycolate. Let us track the dynamic digestion and absorption process. When fat enters the small intestine, let's start from here, it forms large hydrophobic fat globules. Because these globules hide their lipid molecules internally, That means the lipid molecules are hidden inside these globules. Let's say these are the lipid molecules we're talking. These hidden lipid molecules cannot be accessed by pancreatic lipase. To overcome this, we introduce the process of emulsification, which is strictly physical and it's not a chemical change. The amphithetic bile salts surround the large fat globules. This globule we're pointing at, and the bile salts act like detergents to physically break it apart into tiny droplets, and this yellow fat globule is broken down into tiny droplets here, and as a result the surface area increases, and this increased massive surface area for lipase allows pancreatic lipase to efficiently digest triglycerides into free fatty acids and monoglycerides. This blue Pac-Man-like structures are pancreatic lipase, and this tiny droplet is further broken down into smaller pieces, i.e. free fatty acid and monoglycerides. Once these lipolytic cleavage products are freed, we establish the assembly of micelles, which act as the essential nutrient ferry. These aggregates form tiny spheres measuring 4 to 8 nanometer in size, structurally organized with an outer hydrophilic phospholipid shell and a hydrophobic lipid core containing the digested lipid products and bile salts. These micelles solubilize the hydrophobic fats and fat-soluble vitamins, specifically vitamins A, D, E, and K, and ferrying them through the aqueous environment of the intestinal lumen to diffuse directly into the brush border. This is the brush border, or you can say microvilli. After diffusing inside the brush border, the lipid components are released for enterocyte absorption. So parallelly, we showed how food was going from the stomach to intestine and the microscopic structural change that we observed in this sequence of diagram. Following nutrient absorption, we map out the recycling loop and clinical impacts. The body does not waste these valuable bile salts or cholesterol derivatives. Instead, the enterohepatic circulation recovers most bile salts in the terminal ileum. We mean the last portion of small intestine. From the ileum, these salts travel through the portal vein. This is the progression of the travel. The bile salts, as we were saying, travel through portal vein back to the liver to be integrated into bile again. This recycling system operates at a 95% recovery rate per cycle, meaning only 5% is lost in feces, which is regularly replenished by new synthesis in the liver. If this pathway is interrupted, we observe the severe consequences of biliary obstruction, leading to fat malabsorption, fatty stools known as steatorrhea, and severe fat-soluble vitamin deficiencies. Having mapped out this entire biliary mechanism and recycling loop, we will now transition directly to the explicit text definitions, sequence parameters, and high-yield criteria displayed on our anchor slide, or slide 6. The slide text explicitly defines bile as an amphithetic detergent secretion originating from liver, emphasizing that it is not an enzyme. Amphithetic means it has both hydrophobic and hydrophilic part. Since we know bile acts as a detergent, let's explore how it carries out this emulsification process. This mechanism is defined as the physical breakdown of large fat globules into small droplets, acting to increase the total available surface area for lipase contact. Breaking down these large fat globules allows them to assemble into a specific micelle architecture. Micelles are characterized as 4 to 8 nanometer aggregates configured with a stabilizing phospholipid shell and a central lipid core. And to understand how these micelles form, we need to look at the overall chemical composition profile of bile. The total molecular mixture of bile comprises bile salts, bilirubin, cholesterol, phospholipids, bicarbonate, and water. And within this complex mixture, the key functional components are specific cholesterol-derived bile salt derivatives. The primary functional salts are explicitly listed as sodium perchlorate and sodium glycolate, which are synthesized as direct cholesterol derivatives. The liver continuously synthesizes these derivatives, maintaining steady daily production and storage values. Daily baseline hepatic production is established at 600 to 1000 milliliter per day, which is stored and concentrated inside the gallbladder. And once released from storage, these components launch a highly coordinated absorption and transport sequence. The structural sequence follows a path where lipid products and bile salts assemble into a micelle, then diffuse to the brush border and release the internal lipids for epithelial absorption. As this transport sequence takes place, micelle assembly becomes absolutely critical for vitamin solubilization. The mechanical assembly of micelles is explicitly required to ensure fat-soluble vitamins A, D, E, and K are appropriately solubilized. After completing their digestive job, the body saves these components via the enterohepatic circulation or enterohepatic recycling sequence. The recycling loop relies on a strict anatomical sequence where the terminal ileum reabsorbs bile salts, routing them through the portal vein back to the liver for reuse, and liver resynthesizes bile. And this entire recycling pathway operates under remarkably precise and efficient conservation parameters. The system maintains a high efficiency 95% recovery rate per digestive cycle, leaving a minor 5% to be lost in feces and replenished by hepatic synthesis. Before moving further, let's address the massive high-yield exam traps surrounding this digestive process. Examiners will try to give you a vignette by writing scenarios where bile chemically cleaves covalent bonds in fats. You must remember the trap that bile does not digest fats. Lipase does the chemical work. Bile only physically emulsifies the substrate. Moving past the enzymatic trap, let's analyze what happens during a plumbing failure in the biliary obstruction syndrome. Watch out for questions that isolate fat malabsorption from vitamin deficiencies during a plumbing failure. The text highlights that a bile duct obstruction always triggers a dual clinical deficit. First is severe fat malabsorption manifesting as steatorrhea and a systemic deficiency of vitamins A, D, E, and K. Let's wrap up by diving into high-yield exam pearls you must memorize for the exams. A clinical result of severe vitamin K deficiency from biliary obstruction is the development of coagulopathy, where a lack of vitamin K leaves clotting factors unactivated, leading directly to prolonged prothrombin time and clotting failure. And then, when biliary obstruction impairs the micellar transport of vitamin D, it causes impaired calcium absorption across the intestinal epithelium, as calcium uptake pathways are heavily dependent on vitamin D activity. And because the 95% recovery rate of bile salts relies strictly on specialized active transporters located exclusively within the terminal epithelium of ileum or terminal ileum, surgical resection or inflammation of this specific intestinal segment causes global bile acid depletion and subsequent fat malabsorption. The pancreas is a gland that does two completely different jobs in two complete different ways, and they share the same physical organ. Understanding this duality is essential because the IMAT will ask about both, sometimes in the same question, and confusing them is a reliable way to lose marks. This is duodenum, and we position the pancreas, this structure, nestled within the loop of the duodenum, tracing its main pancreatic duct, this one, alongside superiorly located hepatic artery and portal vein. We add the common bile duct, this one, which merges with the main duct at the orifice of common bile duct and pancreatic duct, this place, which empty into the duodenum. Let us separate the organ into its two core functional zones. The first one is the exocrine pancreas, which forms 99% of the gland. That means if this is pancreas, then the majority portion is involved in secreting enzyme or performing exocrine function. And the endocrine islets, which make up remaining 1%, are scattered throughout pancreas, just like isolated islands, where you will see different kinds of cells such as alpha cell, beta cell, delta cell, PP cell, etc. To get a clear view, now let's pivot to another image. As we were talking about the exocrine pancreas, within this exocrine framework, duct cells secrete 1500 ml of an alkaline buffer daily, which is rich in bicarbonate to neutralize acidic stomach chyme and create an optimal environmental pH for enzyme activity. Conversely, the endocrine islets release metabolic hormones directly into the surrounding capillaries. And if we look close inside the endocrine islets, we will see the alpha cells secrete glucagon to raise blood glucose, and the beta cells secrete insulin to lower blood glucose. And the delta cell secretes somatostatin to inhibit both insulin and glucagon, which are secreted by beta and alpha cells. To observe how the exocrine pancreas safely delivers its digestive tools without self-destruction, we map out the Zymogen cascade. The organ packages its proteases, that means the enzymes which break down proteins, as inactive zymogens, which act as an essential safety fail-safe to prevent organ autodigestion, backed up by active trypsin inhibitors. When the secretion reach the duodenum, they interact with a single villus, which is structured with a thin surface layer, internal blood capillaries, and a central lymphatic lacteal. Let's use a different color. This is the lacteal, which is placed centrally, and this lacteal is anchored to a primary blood vessel loop. Now we will zoom inside this villus to see what's happening there. On the brush border surface layer, we locate enteropeptidase, which functions as the master switch. The activation cascade occurs in a precise three-step sequence. In step 1, the inactive precursor trypsinogen encounters enteropeptidase, and this enteropeptidase converts the trypsinogen into active trypsin. In step 2, active trypsin coordinates a rapid chain reaction acting as the universal activator that cleaves pro-carboxypeptidase. As it's not shown here, we're going to see the image underneath, and you'll see that the trypsin acts as universal activator that cleaves pro-carboxypeptidase into free amino acids alongside other enzymes that we will see in the anchor slide. And in step 3, active trypsin exhibits rapid autocatalytic amplification, cleaving more trypsinogen to drive the cascade forward. And in contrast, if we look at pancreatic lipase, this pancreatic lipase is classified as an active enzyme that requires none of these steps, and this lipase breaks down lipids directly into fatty acids and monoglycerides alongside pancreatic amylase. Now shortly recap the sequence again to have a comprehensive concept. First, the inactive trypsinogen is activated by enteropeptidase, and this active trypsin starts a chemical cascade that further activates other enzymes. And in the final step, this active trypsin undergoes rapid autocatalytic amplification, which activates more trypsin, and the cycle goes on and on like this. Having mapped out this entire structural and mechanical layout, let's jump to the anchor slide. Slide 7 and our previous illustration established that pancreas is divided into a 99% exocrine fraction composed of acinar cells and a 1 to 2% endocrine fraction composed of specialized islets of Langerhans cells, which secrete hormones into blood. The major distinction: exocrine pancreas secretes enzymes and endocrine pancreas secretes hormones. Hormones work via blood in distant organs, and enzymes normally use the path of the duct of the gland and works its magic when it's needed. The exocrine duct cells secrete approximately 1.5 liter or 1500 milliliter of bicarbonate daily to function as an alkaline buffer, neutralizing stomach acid to optimize enzyme activity. While the duct cells manage fluid volume, the endocrine matrix handles critical systemic hormone secretion. The endocrine islets house a highly scannable subcomponent matrix where beta cells secrete insulin, alpha cells secrete glucagon, and delta cells secrete somatostatin, all directly into the blood. And we already have said insulin lowers the blood sugar and glucagon does the opposite. And this delta cell works as a controller of both alpha and beta cells by secreting the hormone somatostatin. Or to be more precise, somatostatin is an inhibitor. And fun fact, this hormone universally inhibits other glands or the action of hormones. That's why somatostatin is sometimes called universal inhibitor. Let's follow the precise structural pathways these distinct secretions use to leave the pancreas. Secretions move through the pancreatic duct, this is the pancreatic duct, and also accessory pancreatic duct, this one, toward the duodenal orifice, this orifice we're talking. And by this way, the juices carried over by the ducts of pancreas come over here. And once these juices hit the duodenum, the vital Zymogen activation sequence officially begins. The activation mechanism initiates as the duodenal brush border enzyme enteropeptidase leaves the inactive precursor trypsinogen into active trypsin. And now that active trypsin is generated, it triggers a massive downstream cleavage wave. This active trypsin drives the universal cleavage sequence, transforming chymotrypsinogen, proelastase, procarboxypeptidase A and B, procolipase, and phospholipase A2 into active proteases and lipolytic helpers. This cascade moves rapidly because trypsin fuels its own exponential production, and that is called autocatalytic amplification because generated trypsin continuously cleaves remaining trypsinogen molecules. Thus, by revisiting the diagram, we have completed step 1 and step 2. Also step 3 that we addressed as autocatalytic amplification. And to keep this aggressive autocatalytic system from destroying local tissue, built-in safeguards exist. Autodigestion is actively prevented by packaging enzymes as inactive zymogens and providing protective trypsin inhibitors in the juice as a safety fail-safe. While proteases require safe inactive packaging, other vital enzymes are secreted fully functional. Pancreatic amylase and pancreatic lipase are explicitly categorized as active without activation, meaning they are not zymogens upon luminal release. Now pay critical attention to these high-yield exam traps below. Examiners frequently will try to catch students off guard by claiming that all pancreatic enzymes are secreted as inactive zymogens. You must protect your score by remembering the trap that pancreatic amylase and pancreatic lipase are secreted completely active. Only the protease are released as zymogens. And watch out for questions that state enteropeptidase directly activates procarboxypeptidase or proelastase. The explicit trap is that enteropeptidase activates trypsinogen only, and it is trypsin that then activates all other proteases in the duodenal lumen. Let's wrap up by strengthening the absolute highest yield exam pearls you must memorize for your exams. The rapid autocatalytic amplification of trypsin represents a major clinical vulnerability. If trypsinogen is prematurely converted to active trypsin within the pancreatic duct instead of the duodenum, it overwhelms the local trypsin inhibitors and triggers acute pancreatitis via organ autodigestion. Then we can talk about bicarbonate as well. The production of 1500 ml of daily bicarbonate by duct cell is a mechanism required for small intestinal function because pancreatic proteases, amylase, and lipases are entirely denatured and rendered non-functional if the acidic pH of gastric chyme is not instantly neutralized. And because the master switch enteropeptidase is restricted to microvilli of intestinal villus. This is the villus, and we have enterocytes over here, and over enterocyte we have brush border. The enteropeptidase is restricted to the microvilli only. And for that, the human body ensures aggressive protein splitting activity is completely absent from the upper tracts, preventing premature mucosal erosion. This slide is a complete reference table for pancreatic enzymes. In the exam, you will be given a substrate and asked which enzyme acts on it, or given an enzyme and asked what it produces. You need this table code. Let me walk you through each row and give you the reasoning, not just the fact. This table will outline the activation rules, substrate, and resulting products for the pancreatic enzymatic cascade. Let's start building this matrix by analyzing how trypsinogen initiates the entire cascade. The proenzyme trypsinogen is activated by enteropeptidase and accelerated via autocatalysis, targeting polypeptides as its substrate to yield smaller peptides. Once active trypsin is generated, it immediately turns around to activate chymotrypsinogen. Chymotrypsinogen is cleaved and activated directly by trypsin, selectively targeting aromatic amino acid bonds to break down substrates into smaller peptides. Alongside chymotrypsin, trypsin also targets proelastase to break down dense connective proteins. Proelastase is transformed by trypsin into its active form, breaking down elastin and peptides to yield smaller peptides. While these enzymes target internal bonds, this pathway, called procarboxypeptidase A/B pathway, breaks down terminal protein chains. Uniquely among the primary proteases, procarboxypeptidase A/B is activated by trypsin to act specifically on C-terminal amino acids, yielding free amino acids rather than shorter peptide chains. Shifting gears from proteins, let's look at how pancreas processes dietary fats. Pancreatic lipase is secreted in an active state but strictly requires co-lipase to process triglycerides, yielding 2 monoglyceride and 2 fatty acids. Now let's look at how pancreas processes the unbroken portion of polysaccharides. And for that, pancreatic amylase comes into play. This amylase is fully active upon release with no activation required, digesting starch and glycogen into maltose and dextrins. Returning to fats, this phospholipase A2 pathway specifically targets the structural lipids of cell membrane, and it's activated by trypsin. By targeting phospholipids, it yields lysophospholipids and a free fatty acid. To round out lipid digestion, this profile targets complex dietary cholesterol ester. We're talking about cholesterol esterase. Cholesterol esterase is secreted in a fully active state, breaking down cholesterol esters into free cholesterol and fatty acid. Examiners routinely build tricky questions around the specific end products of these proteases. You must remember the trap that trypsin, chymotrypsin, and elastase are endopeptidases that only yield smaller peptides. Only the exopeptidase, carboxypeptidase, cleaves C-terminal amino acids to yield true free amino acids. So what I was saying is that to get completely free amino acid, you would need exopeptidase, such as carboxypeptidase. Beyond proteins, a classic trap focuses on whether pancreatic lipase is secreted active. The slide highlights the explicit trap that pancreatic lipase is secreted active. We have seen it in the box, but its functional limitation is that a co-lipase helper is required to permit substrate access at the lipid droplet surface. And that's written in the example section, that co-lipase anchors lipase to emulsion droplet surface and activated by trypsin from procolipase. And also watch out for questions trying to make you activate pancreatic amylase prematurely. It is entirely active with no activation required, processing complex carbohydrates into intermediate dimers and oligomers immediately upon entering the duodenal lumen. That means the amylase is secreted active entirely. And another thing I want to mention is the structural transition from Zymogen to active enzyme serves as a localized protective mechanism because trypsin acts as a master biochemical switch for chymotrypsin, proelastase, procarboxypeptidase, and phospholipase A2. Any premature duodenal switch on causes immediate mucosal damage. And finally, to wrap things up, we can talk about pancreatic lipase, lipid cleavage chemistry. Pancreatic lipase displays narrow structural specificity during lipid down packaging, leaving the primary ester bonds of triglyceride molecule at position 1 and 3 to yield exactly 1, 2 monoglyceride and 2 free fatty acids as final products. We have arrived at the centerpiece of the entire digestive system, the small intestine, where the vast majority of nutrient digestion is completed and where almost all of it is absorbed. The small intestine is approximately 6 to 7 meters long in a living person. That length is necessary because the process of absorption requires time and surface contact, and both of those scale with length. To understand how the gastrointestinal tract transitions from a chemical reactor into an absorption powerhouse, we build our concept with the help of our detailed infographics, one segment at a time. We structured this system by dividing its length into 3 specialized anatomical zones. First, we introduce the duodenum, representing the first 25 centimeter of the canal, which functions primarily as a high-yield mixing chamber. Within this initial segment, incoming gastric chyme mixes dynamically with bile and pancreatic juice, while specialized Brunner's glands secrete alkaline mucus to neutralize stomach acid. Now that we have neutralized the acidic transit, the narrative flows organically into the next structural zone where the absolute bulk of breakdown and capture occurs. We position the jejunum, measuring approximately 2.5 meters in length, and this jejunum establishes itself as the primary absorption hub for carbohydrates, proteins, and vitamins. As the remaining contents stream past this hub, we need a natural neural bridge to guide us to the final downstream segment. Having processed the majority of nutrient groups, the canal requires a dedicated zone to secure remaining high-value compounds before entry into large intestine. For this purpose, we locate the ileum at the terminal end, spanning 3.5 meters in length, which is heavily specialized for final recovery, actively absorbing vitamin B12 and recycling bile salts for the enterohepatic circulation. To accommodate this massive functional demand, the small intestine utilizes structural modifications to achieve a staggering 600 times surface area expansion. Let us build this anatomical magnification hierarchy from the largest macroscopic structures down to the cellular level. On the base layer of the intestinal wall, we place circular folds, alternatively known as plicae circularis, which are visible macroscopic folds providing an initial 3x increase. To expand on its architecture, we add finger-like mucosal projections you can see in the second segment, which are called villi, onto these circular folds, and they measure 0.5 to 1.6 millimeter tall and deliver a further 10 times surface area increase. Each individual villus contains an internal capillary network. Let's say this villus we're talking about, if we look inside it, you will see capillary network, artery, and vein at the same time, and the lymphatic duct that is inside this villus, we call it lacteal because it looks white when the fat stays inside that. Finally, looking directly at the apical surface of the enterocytes covering this villi, we introduce microvilli, which form a 1 micrometer microscopic projection border known as the brush border. This is the brush border that we want to refer to, and it finally provides a final 20x amplification that serves as the precise site of final digestion and absorption. Now that we have established this sprawling microscopic border, we must look closer at the specific biochemical gatekeepers anchored directly within this brush border to see how monosaccharides and proteins are processed. We position membrane-bound brush border enzymes alongside specialized transport proteins in this particular image segment. When carbohydrates arrive, let's say maltase has arrived here, it cleaves into glucose plus glucose. Remember, whenever you will add the suffix ase, it will become enzyme. For example, when you say maltose is the carb we're talking about, but when we say maltase, it becomes the enzyme that cleaves maltose into glucose. And just like that, sucrase splits sucrose into glucose plus fructose, and lactase cleaves lactose into glucose plus galactose. Additionally, isomaltase, also known as alpha-dextrinase, breaks down the alpha-1,6 bonds in dextrins to yield glucose. And for protein processing, aminopeptidases target N-terminal peptides to yield free amino acids. When we say N-terminal, this means the starting point of a peptide chain. Nothing to be confused about that wording. And as we were saying, aminopeptidases target N-terminal peptides to yield free amino acids, while dipeptidases cleave dipeptides into free amino acids. Now let us map out the precise transport dynamics of monosaccharide transport across the enterocyte membrane. This is the enterocyte membrane, and we will see how the food particles are transporting across this membrane. Let's start with monosaccharide. First, glucose and galactose move from the lumen side into the enterocyte via SGLT1, a sodium glucose link cotransporter that requires a strong sodium gradient maintained by a secondary active transport. Conversely, we establish fructose as the fructose exception. It moves via GLUT5 through a facilitated diffusion pathway that does not require sodium. Once inside the cell, all three monosaccharides pool near the basal membrane and exit into the blood side via GLUT2 or GLUT3 transporters. Now that we have routed water-soluble sugars into the blood, we must chart the entirely distinct lipid absorption pathways to see how hydrophobic compounds cross this same cellular barrier. Free fatty acids and monoglycerides are delivered to the brush border inside amphipathic 4 to 8 nanometer bile salt micelles. This is the micelle. Upon reaching the membrane, the lipids diffuse passively into the enterocyte. That means it will get inside the cell, intestinal cell or enterocyte. And after getting inside, they are re-esterified into triglycerides and combined with a lipoprotein called ApoB48 inside endoplasmic reticulum to complete chylomicron packaging. This is the chylomicron, our point of interest. Because these large chylomicrons are too bulky to cross blood capillary walls, they follow the lymphatic route. This is the lymphatic route, you see. Through this lymphatic route, the chylomicrons enter the central lacteal to bypass the liver and travel upward via the thoracic duct. However, we must introduce a crucial exception to this lipid rule. The short-chain fatty acid, or SCFA, and medium-chain fatty acids cross the enterocyte membrane independently without micelle packaging or re-esterification, entering the portal blood separately. To be more precise, they directly enter the portal blood system. They don't need any carrier or micelle chylomicron formation. And we have mapped out so many concepts at the same time, and that's why we must look at our anchor slide to solidify our understanding of the whole process. The slide text defines villi as specialized finger-like mucosal projections, where each unit contains a central lacteal and an integrated capillary network. If we zoom in closer on these individual mucosal units, we find the microscopic microvilli brush border. Characterized as the brush border, microvilli are positioned directly on the apical enterocyte surface to serve as the definitive site of final digestion and nutrient absorption. When we say apical surface, it simply means if this is the intestine and we have villus like this, and the microvilli will be toward the luminal or apical site, or facing the lumen can be another interpretation. Together, these villi and microvilli structures combine to drive massive surface area amplification scales. The structural layout outlines a highly scannable hierarchy within this box, where circular folds or plicae circularis, which are visible to the naked eye, provide a threefold increase, and villi measuring 0.5 to 1.6 millimeter provide a 10-fold increase, and microvilli measuring roughly 1 micrometer provide a final 20-fold increase, culminating in a total 600 times surface area expansion. And this specialized surface area is distributed across 3 distinct zones within our regional intestinal framework. The macroanatomy transitions through 3 specialized zones: the 25 centimeter duodenum, where chyme, bile, and pancreatic juice mix near Brunner's glands; the 2.5 millimeter jejunum, which works as a hub for maximizing carb, protein, and vitamin absorption; and the 3.5 meter ileum, dedicated to vitamin B12 and enterohepatic bile salt recycling. As nutrients travel through these zones, complex sugars meet the highly specific carbohydrate brush border matrix. The enzymatic sequence maps out maltase, converting maltose to glucose plus glucose, that means 2 glucose molecules; lactase, splitting lactose to glucose and galactose; and sucrase, doing glucose and fructose; and isomaltase, aka alpha dextrinase, cleaving alpha 1,6 bonds in dextrins to glucose. Alongside sugar breakdown, the mucosal wall utilizes a specialized peptidase and activator matrix for proteins. Protein processing is driven by aminopeptidases, converting N-terminal peptides to free amino acids. We already mentioned N-terminal means the starting point of the peptide branch. Whenever you see C-terminal, that means the terminating point of the chain. And now dipeptidase, it will convert dipeptides to free amino acids, and enterokinase, converting inactive trypsinogen into active trypsin. And once these enzymes break down nutrients, the single sugars rely on transporter mapping. There are different transporters we have discussed, such as SGLT1, GLUT5, GLUT2, and GLUT3. Just keep them in mind. You don't need to memorize them verbatim. And another thing I want to mention is lipid transport mechanism again. Unlike water-soluble sugars, fat digestion products require completely different lipid transport mechanisms. Hydrophobic lipids utilize micelle delivery to diffuse into the enterocyte, then undergo chylomicron packaging alongside ApoB48, and they follow the lymphatic route through the central lacteal to thoracic duct, while short-chain and medium-chain fatty acids serve as the SCFA and MCT exception by directly entering the portal blood. SCFA stands for short-chain fatty acid and MCT stands for medium-chain fatty acids. Now let's switch gears to the major high-yield exam traps testing this absorption mechanics. Remember the trap that lactase intolerance is usually a relative deficiency. Symptoms like osmotic diarrhea and bloating arise when undigested lactose remains in the lumen, drawing water into the colon where bacterial fermentation of lactose takes place. Osmotic diarrhea happens when your body cannot digest certain foods or drinks, and these undigested substances stay in your bowels and pull extra water into your intestines, making poop loose and watery. And a fun fact about this type of diarrhea is that it stops when you fast or stop eating. Now let's lock in the highest yield must-know pearls to maximize your score. One is written, and I will give some more pearls for your exams. The first one talks about the vessel boundary. As established in the pearl, the structural destination of a nutrient depends heavily on its biochemical class. The central lacteole, this is the villus, and in center we have lacteole, drains exclusively to the lymph network to carry large chylomicron fats, while the blood capillary, parallel to this lacteole, drains directly to the portal vein to route water-soluble nutrients straight to the liver. Then there would be secondary active prerequisite. Glucose absorption via SGLT1 is fundamentally dependent on the basolateral sodium-potassium ATPase pump. Basolateral pump is a tiny pump found on the bottom sides of the cells that line your intestines and kidneys. Let's say this is a segment of intestine and we have cell. This side is luminal side or apical side, and this side over here is basolateral side, and the pump will be here on the basolateral side. That's why it's called basolateral pump. And this pump works on the sodium gradient. Whenever this gradient is disrupted, the secondary active transport collapses, causing severe malabsorption despite normal luminal glucose concentration. That means there can be presence of malabsorption even after the glucose being normal. The glucose concentration. Then the pearl that is written here, it talks about the thoracic duct bypass. Because chylomicrons enter the lymphatic system via lacteoles rather than blood capillaries, absorbed dietary lipids completely bypass the hepatic portal circulation and liver metabolism, entering the systemic venous blood directly via thoracic duct. Now that you know the structural amplification strategy the small intestine uses, let us travel through its three regions and understand what each one specializes in. Small intestine, as I said earlier, is not a homogeneous tube. The duodenum, the jejunum, and ileum have a distinct identity, a distinct structural feature, and a distinct absorptive specialization. The IMAT tests this regional specificity repeatedly, particularly for vitamin B12 and bile salt absorption. We have covered all these absorption topics in details in our previous segments. That's why we will now focus mainly on the transporters in this phase. It might feel redundant to you at times that I explain the same mechanism in different angles, but that's what will give you true mastery on a particular topic. We establish the whole system within the cell wall of an enterocyte. Let's start looking from the left side. This one is an enterocyte. This is the nucleus. And this enterocyte separates the intestinal lumen at the top from the blood capillary at the base. At the lower boundary, we locate the basolateral membrane. This is the basolateral membrane, and we place a critical energy engine directly within it. This is the engine we are talking about. This is called the sodium-potassium ATPase pump. This active transporter continuously pumps sodium ions out of the cells into the bloodstream while moving potassium ions inward, utilizing adenosine triphosphate to maintain a steep extracellular sodium gradient. Now that we have established this driving sodium gradient at the base of the wall, we can transition naturally to the apical membrane. This is the apical membrane, which faces the gut lumen to see how carbohydrates exploit this energy. We position the specialized co-transporter SGLT1 on this apical border. When glucose and galactose arrive in the lumen, they bind to SGLT1. These are the transporters, and when they are bound, the transporter will pull these monosaccharides. Let's say one glucose has arrived here, and this glucose will be pulled by the transporter across the apical membrane via secondary active transport coupled with the downward movement of sodium. That means both glucose and sodium will get inside the cell. As sodium gradient plays a role in this glucose transport, we call this secondary active transport mechanism. However, the cell handles different carbohydrate classes through entirely separate pathways. And for that, we come to the fructose exception. We position the GLUT5 transporter right next to SGLT1 to handle fructose entry via facilitated diffusion. We position the GLUT5 transporter as a green color-coded transporter here. Unlike other sugars, this channel has a slower absorption rate and does not require sodium or ATP once all three monosaccharides pull inside the cytoplasm. The first being glucose, then being galactose, and then fructose. They require a shared exit route to reach the systemic circulation. Their target is to come inside the blood vessel. And for that specific purpose, we introduce some other transporters. We have 3 transporters here, and they are all the same, and they are called GLUT2 transporters. And they are situated on the basolateral membrane of the enterocyte. And they act as a universal gateway, allowing glucose, galactose, and fructose to exit into the surrounding blood capillary. This is the blood capillary, and the 3 monosaccharides get inside this blood capillary via passive facilitated diffusion. Having resolved carbohydrate capture, we must look at how the enterocyte handles nitrogenous compounds, moving organically into the adjacent protein and peptide absorption pathways. That means now we shift our focus to this segment of the process. We map out 2 distinct entry gates on the apical membrane to handle the products of proteolysis. First, we establish sodium-dependent co-transporters to move free amino acids. These are the free amino acids across the apical membrane, actively coupling their entry to the primary sodium gradient before they exit the basolateral membrane into the blood pool. I want to remind you again, this membrane is the apical membrane. This side represents basolateral, and this is the blood capillary for better context. For larger fragments, we introduce a completely distinct transport system located on the apical membrane, which is called the PEP-T1 transporter. This channel imports small dipeptides or tripeptides intact into the cell. Let's say these are the dipeptides or tripeptides. They will get inside the cell through this transporter. And this movement is driven or powered by hydrogen ion gradient, or simply proton gradient. Once inside the enterocyte, the small peptides must be broken down into individual building blocks before entering the blood, initiating a process of intracellular peptide hydrolysis. We add cytoplasmic peptidases within the interior of the cell, which rapidly cleave the absorbed di and tripeptides into free amino acids rather than leaving them to be digested in the lumen, ensuring only monomers enter the blood capillary. Now let me quickly remind you about the position of duodenum, jejunum, and ileum. Duodenum is 25 centimeters in length, and its specialization is around Brunner's gland, producing alkaline mucus to mix the bile and pancreatic enzymes, resulting in heavy enzymatic digestion but minimal absorption. So duodenum stands for maximal digestion and minimal absorption. And once the chyme is fully digested and neutralized, the narrative flows naturally into the primary absorption center of the tract, that is the jejunum, measuring 2.5 meters in length. They have highly specialized villi and maximum brush border enzyme activity to absorb the bulk of your glucose, amino acids, fatty acids, minerals, and most vitamins. Finally, the remaining unabsorbed material must be cleared by a terminal recovery zone before entering the large intestine, leading us to the final segment, the ileum, which is 3.5 meters in length. They have shorter villi, they have specialized transporters such as ASBT, not that important, you can discard it. And also, ileum has intrinsic factor IF B12 receptors to execute the final recovery of bile salts and vitamin B12 before reaching the ileocecal valve. Ileocecal valve means the valve that is situated between the ileum and the first part of large intestine, called the cecum. As we understand how the molecular nutrients traverse the epithelial barrier, we can scale up our perspective to examine how the microscopic regions of the small intestine divides these responsibilities. We map out the 3 sequential segments of the tract, beginning with the hepatopancreatic ampulla, or ampulla of Vater, where the shared bile and pancreatic duct. This is the bile duct and this is the hepatopancreatic duct, and both of the ducts open into the duodenum under the direct control of the sphincter of Oddi. We are done mapping everything successfully. Now let's get back to our anchor slide for a quick recap. First things first, managing the entire flow of food requires upstream regulation at the main anatomical junction. The hepatopancreatic ampulla or ampulla of Vater represents the shared bile and pancreatic duct opening into the duodenum, regulated directly by the sphincter of Oddi. As we have covered the cellular mechanisms in detail, let us trace these events across specific anatomical zones. The duodenum measures 25 cm in length, specializing in alkaline mucus from Brunner's glands alongside bile and pancreatic enzymes to focus on enzymatic digestion with minimal absorption. After mixing in the duodenum, the nutrient slurry enters the main absorptive site. Spanning 2.5 meters, the jejunum utilizes tall villi and maximum brush border enzymatic activity to absorb glucose, amino acids, fatty acids, most vitamins, and minerals. Spanning 3.5 meters, the ileum features shorter villi, the active ASBT transporter, and IF-B12 receptor complex to absorb bile salts and vitamin B12 before the ileocecal valve. That means ileocecal valve is not a part of cecum, it's a part of ileum. To know about cecum, you gotta study large intestine. Now let us apply these anatomical concepts to common pitfalls found on the exam. Examiners frequently try to trap students by stating that vitamin B12 can be absorbed fluidly across any section of the small intestine. You must remember the trap that B12 is absorbed only in the terminal ileum via the IF receptor. Any distal resection, that means if you remove distal part of small intestine, it will lead directly to a total halt in absorption and it will cause pernicious anemia as a result. Similarly, examiners love to test the highly localized recycling of the bile salts. Watch out for questions that confuse the active transport of bile salts with passive lipid diffusion. The slide marks the explicit trap that bile salt reabsorption happens only in terminal ileum via the active ASBT transporter rather than passing through the upstream jejunum. Let us conclude by reviewing the highest yield clinical takeaways for the test day. Established in the canvas phase and traps as surgical terminal ileum resection. That means if you surgically remove the terminal ileum, it triggers a devastating dual clinical deficit, causing both a systemic vitamin B12 deficiency and a rapid depletion of the circulating bile salt pool, which results directly in fat malabsorption and steatorrhea. This slide 11 reinforces the transport mechanisms of different nutrients, and we have covered everything in canvas phase. So we will directly review this slide without any visual aid. First things first, the slide text defines the secondary active transport mechanism as being driven by ion gradient. To be more precise, sodium ion gradient, where ATP is not directly used by the transporter itself, and facilitated diffusion is characterized as a carrier-mediated, energy-independent transport pathway that moves solutes down their concentration gradient. The structural layout or this box maps out glucose and galactose, both utilizing apical SGLT1 via secondary active transport or secondary active sodium transport, and fructose being an exception using the GLUT5 transporter and the mechanism being facilitated diffusion. But the basolateral exit, as we have said earlier, all of the three monosaccharides will use the same transporter to move from the basolateral site to the bloodstream. Now coming to the amino acid part. Three amino acids rely on a sequence transitioning from apical sodium dependent co-transporters to basolateral transporters, dumping directly into the portal blood. Dipeptides or tripeptides utilize apical PEPT1 transporter mechanism, which is proton dependent, or to be more precise, hydrogen ion gradient dependent, and it moves the di and tripeptides into cytoplasm, where cytoplasmic peptidases leads them into free amino acids, which are ready to be transported to the blood. Both the exam traps and the pearls are your best friends before exam because they will save you lots of scores or marks. Examiners will try to trick you at first by saying that fructose absorption is halted by a lack of sodium or an enterocyte energy deficit. You must remember the trap that fructose uses GLUT5, not SGLT1. It requires no sodium or active energy and exhibits a characteristically low absorption rate compared to other sugars. Also watch out for the second trap. To circumvent this, you need to know SGLT1 or sodium glucose link transporter 1 is powered entirely by the sodium gradient, and it is the sodium potassium ATPase on vasolateral membrane that maintains it. As highlighted in our exam pearls, di and tripeptides are absorbed intact via the specialized PEPT1 channel, but they're fully hydrolyzed into free amino acids inside the enterocyte cytoplasm, not in lumen, and it ensures that only individual amino acid monomers enter the portal blood. This slide again deals with lipid absorption, as lipid absorption is the most structurally elaborate absorption process in the body, and it uses a route that is completely different from every other nutrient. Glucose and amino acids go directly into the portal blood and are delivered to the liver within seconds of absorption. Dietary fat, specifically long-chain fatty acids, takes a detour through the lymphatic system and doesn't reach the liver until it has already entered the systemic circulation. Understanding why this route exists and what happens mechanistically at every step is essential for the IMAT. To fully grasp the complexity of dietary fat processing, we must isolate and build a complete lipid absorption and the chylomicron route from first principles. Our starting point is the intestinal lumen. So this area represents intestinal lumen for us. In this lumen, dietary fats are already digested, with position, micelle delivery, and diffusion near the apical border. This is the apical border. We write AB. These micelles ferry the hydrophobic products to the membrane, where the fatty acids and monoglycerides dissociate from the micelle structure. That means as soon as the micelle comes to the apical border of the membrane, they disintegrate, and the fats, to be more precise, the fatty acids and monoglycerides enter the enterocyte via simple diffusion, an entirely passive process requiring no transporter. Once inside the enterocyte, the free-floating lipids must be processed before they can be moved into the body's aqueous environment. We locate the smooth endoplasmic reticulum, or SER. This is the SER, the whole structure, and establish the process of re-esterification and packaging. Inside ER, these individual lipids are rebuilt into triglycerides. To make these fat droplets soluble in blood and lymph, the cell packages them with an outer shell of phospholipids and a specific structural protein called ApoB48, forming massive lipoprotein particles called chylomicron. Because these newly formed chylomicrons are simply too large to cross the tight junctions of a blood capillary, the cell must utilize a different exit strategy. We locate a secretory vesicle. This is the vesicle we're talking, moving toward basolateral membrane, and establish the process of exocytosis. The chylomicrons exit via exocytosis into the central lacteole. This is the lacteole, which is the central lymph capillary located within each villus. We have mentioned it quite a few times, so I think it's very clear for you now. From the central lacteole, the fluid travels through the broader lymphatics, moving upward through the thoracic duct. That means it will go with thoracic duct, we're writing TD, before finally dumping into the systemic circulation at the left subclavian vein. For that, let's write F, not F, actually S, C, and V. Subclavian vein. This subclavian vein delivers the fats into the blood. Concurrently, we must include the fat-soluble vitamins that are vitamin A, D, E, and K, which follow this exact same path, being absorbed via micelle route and subsequently packaged into chylomicrons. However, biology always represents some exceptions based on molecular size. We map out a distinct alternative pathway known as SCFA and MCT shortcut. This pathway relies entirely on the chain length rule. While long-chain fatty acids containing more than 12 carbons must follow the mandatory chylomicron route through the lymph, short-chain fatty acids, SCFA, those who have carbon number less than 12, and medium-chain fatty acids containing 8 to 12 carbon behave differently. Because these shorter chains are significantly more water-soluble, they bypass the entire re-esterification process and enter the portal blood capillaries, bypassing the lymph systems completely. Okay, we have mapped out the complete lipid absorption and chylomicron route. Now we are ready to pivot to our anchor slide. This slide formally defines chylomicrons as passive lipoprotein particles synthesized by the enterocyte. This slide formally defines chylomicrons as massive lipoprotein particles synthesized by the enterocyte, composed internally of triglycerides and cholesterol, stabilized by an outer layer of phospholipids and the apolipoprotein ApoB48. Now let us look at where these massive lipoprotein particles go after synthesis. Lacteals are classified as the central lymph capillaries located within the core of H. villus, serving specifically to drain large chylomicrons away from the intestinal tissue. Before entering these lymph vessels, lipid must first cross the enterocyte membrane, and the entry sequence is initiated when micelle approaches the brush border, allowing fatty acids and monoglycerides to dissociate from the micelle and cross the apical membrane via passive simple diffusion, where absolutely no transporter is needed. Once inside the cell cytoplasm, the smooth endoplasmic reticulum reveals these lipids. That's why written re-esterifies. After they are re-esterified, they are then packaged as chylomicrons. Next, these packaged chylomicrons leave the cell to enter the bloodstream. The massive chylomicrons undergo exocytosis, entering the central lacteal, following through the lymphatics to the thoracic duct, and finally emptying into the left subclavian vein to join the systemic blood. This same lymphatic pathway also carries vital fat-soluble vitamins. Fat-soluble vitamins A, D, E, and K strictly follow this lipid path, being absorbed via the micelle route and subsequently packaged directly into the core of the chylomicrons. However, the exact route depends entirely on the fatty acid chain length. The biological fate of a fatty acid is dictated by its carbon length. Long-chain fatty acids having carbons more than 12 are forced into chylomicron route via the lymph. Short-chain fatty acids having carbon number less than 12 enter portal capillaries directly because they are water soluble and form no chylomicron. And medium-chain fatty acids having carbon between 8 and 12 also enter the portal blood directly to completely bypass the lymph. Let us expose the common trap regarding short-chain fatty acid transport. Examiners routinely attempt to catch students by stating that all dietary fats are packaged into chylomicrons for transport. You must remember the explicit trap that short-chain fatty acids do not form chylomicrons because of their shorter carbon chains. They're soluble enough to enter the portal blood directly. Another frequent error involves how the enterocyte absorbs the micelle structure. Watch out for questions that describe the entire micelle structure being absorbed intact into the enterocyte. The slide highlights the trap that while the lipid contents diffuse across the membrane, the bile salts remain in the lumen. They are not absorbed with the micelle contents in the jejunum and must travel down to the terminal ileum for recycling. Now let's conclude with the highest yield examples for your exam. As established in our previous canvas, the unique absorption profile of medium chain triglycerides, or MCT, is exploited clinically because they completely bypass chylomicron formation and lacteal entry. They're used as a primary caloric source for patient suffering from severe lymphatic dysfunction or chylomicron packaging disorders. Another extra pearl I want to share is that because the lymphatic system routes all abdominal chylomicrons upward toward the thoracic duct and from there to the subclavian vein, a massive influx of dietary fat can actually turn the plasma of the systemic venous blood temporarily milky white after a heavy meal before it reaches the liver for processing. That's quite interesting to behold. Having absorbed almost everything of nutritional value in the small intestine, this segment is the small intestine. After being absorbed almost everything of nutritional value, the remaining luminal contents enter the large intestine. This is the starting of large intestine, and the connecting point between the large intestine and the small intestine is this valve, which is called ileocecal valve. This valve is a small muscular gate between your small and large intestines that lets digested food pass through while blocking harmful bacteria from backing up. You also can see haustration. Haustration is the bunching up of large intestine into a series of small pouch-like segments. This is one haustration, another one here, and it goes on throughout the whole large intestine. And their job is to slow down the waste to absorb water and form stool. Large intestine is a tube with an approximate length of 1.5 meters. Looking closely at its structural specialization, we establish its microscopic layout by positioning a flat epithelium embedded with deep crypts. These are the crypts. We see 3 of them. The crypts are the deep tube-like valleys that dip downward in the cellular layer or epithelium in our diagram. Unlike the upstream segments of the alimentary canal, we emphasize a distinct structural rule here. This large intestine completely lacks nutrient-absorbing villi and microvilli. Furthermore, we observe that no significant enzyme digestion occurs within this lumen. Now let's discuss its primary physiological functions. The main operational goal here focuses heavily on water and electrolyte recovery alongside compaction. We map out active transport mechanisms across the mucosal membrane that drive sodium and chloride absorption into the tissues. If you look at this segment, you're going to find out. This sodium and chloride absorption is balanced by a secretory pathway that drives potassium and bicarbonate secretion back into the lumen. This aggressive solute transport creates a sharp osmotic gradient that absorbs water, allowing the organ to convert liquid chyme into solid stool. Now let's discuss its primary physiological functions. The main operational goal here focuses heavily on water and electrolyte recovery alongside compaction. We map out active transport mechanisms across the mucosal membrane that drive sodium and chloride absorption into the tissues. If you look at this segment, you're going to find out. This sodium and chloride absorption is balanced by a secretory pathway that drives potassium and bicarbonate secretion back into the lumen. This aggressive solute transport creates a sharp osmotic gradient that absorbs water, allowing the organ to convert liquid chyme into solid stool. However, when we establish a detailed comparison of absorption priorities, we observe a distinct nutrient absorption limit. Unlike the small intestine, which utilizes a massive surface area to maximize the capture of glucose and amino acids, the large intestine absorbs no significant glucose or amino acids, creating a complete cross for standard nutrient groups. While the small intestine handles a high water volume, the large intestine processes a lower volume that remains absolutely crucial for the final dehydration of stool. Now that we have defined boundaries of solute and water recovery, we must explore the incredibly active biological environment shown in this segment. And this segment represents the gut microbiome. We introduce a massive microbial community inside the deep crypts, housing a dense anaerobic ecosystem composed of 100 trillion bacterial cells, which mathematically tracks as an approximate population of 10 to the power 14 bacteria. We position the dominant bacterial classes within this anaerobic population, noting they consist mainly of bacteroids, firmicutes, and bifidobacterium anaerobes. These symbiotic microbes execute a vital biochemical sequence. They ferment complex dietary fiber that human enzymes cannot break down and converts them directly into short-chain fatty acid. And we place the three primary short-chain fatty acids produced by this pathway in our layout. The first one is acetate, the second one is propionate, and the third one will be butyrate. The fermentation byproduct butyrate serves a critical localized role as the primary energy source for the colonocytes, or the cells of the colon, and at the same time for intestinal lining, this one. And also it provides powerful anti-inflammatory protective effects. Concurrently, this microbiome synthesizes essential micronutrients, driving the production of vitamin K2, also known as menaquinone, which is essential for healthy blood clotting factor synthesis, alongside the synthesis of some vitamin B types. We covered everything regarding large intestine, so let's quickly jump to our slides now. The slide formally defines the large intestine as a muscular tube measuring 1.5 meters in length, structurally distinguished by having no villi, containing abundant goblet cells, and executing no significant enzyme digestion. Next, let's explore how this muscular tube physically moves content along its length. The transport sequence relies on a matrix where haustral contractions, as we saw in the diagram or our demonstration, this haustral contraction handle localized segmental mixin, while mass movements execute propulsion to the rectum a few times per day. Beyond moving waste, these specific movements support the organ's core chemical functions. The core functions revolve around water absorption, active sodium and chloride absorption, potassium and bicarbonate secretion, waste compaction, and housing microbiome. The microbiome that is housed by the large intestine plays a critical role for our well-being. And the anaerobic ecosystem, containing 10 to the power 14 bacteria or 100 trillion bacterial cells, consist mainly of the anaerobes, bacteroids, firmicutes, and bifidobacterium, which all of them are anaerobes. This dense bacterial population actively breaks down dietary fiber through metabolic pathways, and the pathway follows a strict sequence where bacteria ferment dietary fiber to yield short-chain fatty acids, specifically producing acetate, propionate, and butyrate. One specific product of this fermentation serves as the primary energy source for colonocytes and the intestinal lining while acting as an inflammatory agent, and that one is butyrate. This metabolic machinery of the gut bacteria also synthesizes critical vitamin K2 or menaquinone to support healthy blood clotting factor synthesis alongside some vitamin B types. After all this discussion, we come directly to critical exam traps. Examiners routinely attempt to catch students off guard by asserting that because the large intestine is highly efficient at absorption, it plays a backup role in capturing leftover simple sugars or protein fragments. You must protect your score by remembering the strict nutrient absorption limit trap. The large intestine does not absorb glucose, amino acids, or fat, with the sole exception of short-chain fatty acids derived from microbial fermentation. Examiners also love to test how common medications disrupt these bacterial pathways. Watch out for clinical scenarios linking broad-spectrum antibiotic to sudden bleeding complications. The text highlights the trap. Heavy use of antibiotics accidentally kills the gut bacteria, which directly leads to a reduced vitamin K12 supply, causing impaired clotting factor synthesis and subsequent clotting failure. Let us conclude by reviewing the highest yield summary concepts for your boards. While large intestine is clinically celebrated for its role in water recovery and compaction, the small intestine actually absorbs significantly higher total water volume daily. The large intestine's unique mechanical job is merely the final conversion of liquid chyme to solid stool. And some bonus pearls are, unlike most tissues in the human body that rely entirely on systemic glucose delivered via the bloodstream, the epithelial cells of the large intestinal lining are unique because they derive the absolute majority of their cellular energy from the local luminal oxidation of butyrate. The absolute transition from the villus-covered mucosa of the terminal ileum to the completely flat epithelium and deep crypts of the cecum serves as a primary diagnostic landmark in histology, confirming entry into a region dedicated purely to fluid balancing and microbial fermentation rather than primary macronutrient capture. Now we come to the segment of hormonal control of the GI tract, or more specifically, we're going to talk about gastrin, secretin, and cholecystokinin. For kind information, the gastrointestinal tract is the largest endocrine organ in the human body. This is a fact that surprises most students who think of the pancreas or the thyroid when they hear endocrine organ. But the GI mucosa contains a staggering array of specialized hormone-secreting cells scattered along its length, collectively producing more distinct hormones than any other organ. And for the IMAT, 3 hormones are absolutely mandatory knowledge. First one is gastrin, then secretin, and CCK or cholecystokinin. You must know where each is produced, what triggers its release, and what it acts on, and what the net physiological effect is. And you must be able to distinguish them from each other when a question gives you a stimulus and ask which hormone is released, or gives you an action and ask which hormone causes it. Now let me give you the conceptual logic before the facts, because the logic makes the facts stick. The GI hormones form a coordinated relay system as food moves from the stomach to the duodenum and triggers and what the net physiological effect is. And you must be able to distinguish them from each other when a question gives you a stimulus and asks which hormone is released, or gives you an action and asks which hormone causes it. Now let me give you the conceptual logic before the facts. The GI hormones form a coordinated relay system as food moves from the stomach into the duodenum, the first part of small intestine. The composition of the chyme changes. Protein arrives first, then fat, and then acid. Each chemical arrives in the duodenum and triggers a different hormonal response tuned specifically to that chemical. Acid triggers secretin, which orders neutralization. Fat and protein together trigger CCK or cholecystokinin, which orders enzyme and bile delivery. And protein in the stomach triggers gastrin, which maximizes gastric acid output before food even reaches the duodenum. This is elegant anticipatory coordination. Gastrin is produced by G cells located in the antrum and pylorus of the stomach. The stimuli for its release are 3. First, protein and peptide fragments in the stomach, distension of the stomach wall by food, and vagal stimulation, arriving during the cephalic phase before food even enters the stomach. Gastrin is released into the bloodstream and acts primarily on parietal cells to stimulate hydrochloric acid secretion. It also acts on chief cells to stimulate pepsinogen release and on the smooth muscle of the stomach to increase gastric motility. The net effect is to maximize the stomach's digestive capacity for protein. Critically, gastrin is subject to negative feedback. When gastric pH falls below about 1.5, somatostatin released from T cells out of the stomach inhibits G cells, shutting off gastrin secretion. This prevents runaway acid accumulation. Secretin is produced by the S cells in the duodenal mucosa. Its single most important trigger is acid, specifically the arrival of H+ ions in the duodenum as gastric chyme enters. You already know stomach terminates at duodenum. So the chyme from the stomach falls into duodenum, and thus duodenum gets flooded with protons or hydrogen ions. Secretin's primary action is on the ductal cells of the pancreas. Secretin stimulates the ductal cells of the pancreas to release large volume of bicarbonate-rich juice. This bicarbonate stream neutralizes the acid chyme and raises the duodenal pH to 6 or 7, which is the range required for pancreatic enzymes to function. Secretin also inhibits gastric acid secretion and slows gastric motility. That means the movement of the gastric smooth muscle, which also slows down the food coming to duodenum, and it buys duodenum some time to neutralize what has already arrived before more acid chyme is delivered. Secretin has essentially one job, and that is acid neutralization. If you see acid in the duodenum in any IMAT question, the hormone you think of is secretin. Then comes CCK, or cholecystokinin, and this is produced by eye cells distributed throughout the duodenal and jejunal mucosa. And the trigger for this hormone, or triggers, are fat and protein arriving in the duodenum. CCK has the most diverse range of actions of the three hormones. It causes contraction of the gallbladder, ejecting concentrated bile into the duodenum. It stimulates the acinar cells of the pancreas to secrete their full enzyme package. It relaxes the sphincter of Oddi, opening the shared duct so both bile and pancreatic juice can flow. It inhibits gastric emptying, slowing the rate at which more chyme arrives from the stomach, giving the enzymes already in the duodenum time to complete their work. And it acts on the hypothalamus, part of our brain, via vagal nerve to induce satiety or fullness. And that is why a fat-rich meal is more satiating than a carbohydrate meal of the same caloric content. The entirety of the table is decently covered in our canvas phase, and we started the canvas phase by saying GI tract is the largest endocrine organ in the body. And now you have to watch out for the exam traps. The first one is gastrin is inhibited by its own product. Never get confused when these types of statements are given to you. When HCl production lowers the gastric pH below 1.5, somatostatin from D cells inhibits G cells, switching off gastrin secretion. This negative feedback prevents the stomach from acidifying itself to destruction. On IMAT questions about gastrin regulation, always note this self-limiting feedback. Also remember that gastrin stimulates both acid secretion and gastric motility. Students who learn only the acid stimulating effect miss the motility component and will get questions about gastric emptying wrong. Second trap states that secretin is released only by acid. CCK is released by fat and protein. Do not mix this up. An IMAT question that says fat-rich chyme enters the duodenum is asking about CCK, not secretin. A question that says acidic chyme enters the duodenum is asking about secretin. The stimuli are completely distinct even though both hormones act on pancreas. When talking about this exam pearl, we will focus on CCK again. CCK is the molecular explanation for why fatty meals feel more filling than carbohydrate meals of the same caloric value. CCK acts via vagal efferent pathways to signal the hypothalamus, reducing hunger. It also slows gastric emptying so food stays in the stomach long. Both effects contribute to the extended satiety after a fat-rich meal. This is tested in IMAT questions that cross over between GI physiology and appetite regulation. We have been discussing hormonal control, which is the chemical broadcast system of the gut, where one cell releases a hormone, it travels through the blood, and it acts on target cells far away. But nervous system or nervous control operates differently. It is faster, more localized, and more directly tied to the moment-to-moment mechanical state of the gut. The gut has two levels of nervous control. Its own intrinsic nervous system called the enteric nervous system and extrinsic inputs from the autonomic nervous system. The enteric nervous system, or ENS, is embedded within the walls of the gut from the esophagus to the rectum. It contains approximately 100 million neurons, roughly the same number as in the spinal cord. It can orchestrate complete digestive behaviors, including peristalsis, segmental contraction, secretion, and blood flow regulation, entirely independently of any input from the brain or spinal cord. This is why the ENS is called the second brain. It is not metaphor. It is a genuinely autonomous nervous network. And just as recap, we can remind ourselves that ENS is organized into two plexuses. First one is Auerbach's or myenteric plexus, sits between the inner circular and outer longitudinal muscle layers of the muscularis externa. It primarily controls smooth muscle activity, meaning it coordinates the peristaltic waves and segmental contractions that propel and mix gut contents. Meissner's plexus, formerly the submucosal plexus, lies in the submucosa and primarily regulates secretion and blood flow. If you cut the vagus nerve of an experimental animal, the animal's gut continues to produce peristaltic waves. The ENS handled that independently. What the vagus nerve does is modulate the ENS, amplifying or dampening its activity depending on the body's overall state. After we are done with the enteric nervous system, we pivot to autonomic nervous system. The autonomic modulation is divided into parasympathetic and sympathetic nervous system. The parasympathetic nervous system, conveyed primarily by vagus nerve, which is cranial nerve number 10, promotes all aspects of digestion. Vagal activation increases salivary secretion, stimulates gastric acid and enzyme secretion, increases intestinal motility, and relaxes the sphincter to keep the contents moving. This is the rest and digest state. When you sit down to a relaxed meal, your parasympathetic system dominates, and every gland and smooth muscle in your gut is primed for maximal digestive efficiency. Now, the sympathetic nervous system does the opposite. It is the fight or flight system when you are under threat or intense stress. The sympathetic division shuns blood to the skeletal muscles. As sympathetic nervous system shuns blood away from the gut to skeletal muscles, the blood supply reduces to GIT. The sympathetic division also dilates the pupil, accelerates the heart, and suppresses everything that is not immediately necessary for survival. That's why it's written sympathetic nervous system is fight or flight response generator. Digestion is not necessary for immediate survival. So sympathetic activation reduces intestinal motility, constricts digestive sphincters, and decreases secretion. This is also why stress consistently worsens gut conditions like IBS or irritable bowel syndrome. Now, the sequence of phases of digestion. Digestion is divided into 3 sequential but overlapping phases named for where the controlling stimuli originate. The first one is the cephalic phase. This cephalic phase begins before food even enters your mouth. The sight, the smell, thought, or taste of food activates sensory centers in the cerebral cortex, which relays signals to the hypothalamus, which activates the vagus nerve. The vagus then drives salivary secretion, gastric acid secretion, and pepsinogen release. This anticipatory preparation is why your mouth waters and your stomach begins working before you take the first bite. It is a preparatory response that ensures the digestive machinery is running by the time food actually arrives. The second phase is the gastric or stomach phase. This phase begins when food enters the stomach. 2 stimuli drive it. The first one is mechanical distension of the stomach wall detected by stretch receptors that activate vagal reflexes and local ENS circuits, and the presence of protein and peptide in the lumen, which directly stimulates G cells to release gastrin. The gastric phase produces the largest volume of gastric secretion and the greatest increase in the gastric motility of the 3 phases. The intestinal phase begins when chyme enters the duodenum. This phase has both stimulatory and inhibitory components. The arrival of nutrients stimulates local secretion of digestive juices by the intestinal mucosa, but the arrival of acid, fat, and hypertonic material also triggers feedback signals that slow gastric emptying. The enterogastric reflex, mediated partly by secretin and CCK or cholecystokinin by direct sympathetic nerve signals. The intestinal phase therefore simultaneously promotes intestinal digestion while breaking the stomach to prevent it from delivering chyme faster than the duodenum can handle. We completed everything in our canvas phase. Now we need to move to the anchor phase to recap what we have studied and the important caveats for exam. The slide formula defines the enteric nervous system, or ENS, as an autonomous neural network consisting of two highly specialized plexuses that functions entirely without direct input from brain or spinal cord. Nestled within this independent network, Auerbach's plexus, also termed myenteric plexus, is located between the circular and longitudinal muscle layers to serve as the primary driver coordinating peristalsis. Now that we have established the muscular motor drive, the neural architecture shifts to a separate localized network to manage glandular outputs. Meissner's plexus, alternatively designated as the submucosal plexus, is situated directly in the submucosal layer to regulate gastrointestinal secretion and local blood flow. While these two internal plexuses manage the baseline activities of the tract, their functions are continuously modulated from the outside by autonomic nervous system to match shifting behavioral states. That's why the next title is Key Features Autonomic Modulation. The parasympathetic division, driven by the vagus nerve or cranial nerve 10, orchestrates the classic rest and digest response, which acts to stimulate overall motility and secretion while relaxing regional sphincters. Conversely, the body handles stress states by flipping an entirely opposite autonomic switch to shut down these digestive workflows. The sympathetic division coordinates the fight or flight response, which operates in direct opposition to vagus nerve by inhibiting motility, constricting sphincters, and reducing total secretion. This coordinate interplay between internal plexuses and autonomic inputs guides the system through a strict chronological timeline, mapping out the three phases of digestion. The sequence initiates with the cephalic phase, where the mere sight, smell, or thought of food sends signals through the hypothalamus to vagus nerve, triggering anticipatory saliva and HCl secretion. Once the physical bolus actually crosses the gastroesophageal junction, the presence of material shifts the mechanical and chemical timeline to the next zone. The gastric phase takes over as food in the stomach causes mechanical distension and introduces protein, stimulating G cells to release gastrin, which drives heavy hydrochloric acid and pepsinogen production. Finally, as this processed material leaves the gastric reactor to enter the small intestine, the tract activates its last major regulatory sequence to coordinate downstream processing. The intestinal phase is triggered when chyme arrives in the duodenum, causing the release of secretin and cholecystokinin to stimulate a massive delivery of neutralizing pancreatic juice and emulsifying bile. Now pay critical attention to the traps. Examiners frequently will try to trick students by asserting that the sympathetic nervous system stimulates gastric secretions to help deal with stress. You must remember the explicit trap that vagus nerve stimulates gastric acid secretion because it is parasympathetic, while the sympathetic division actively inhibits it. Watch out for clinical questions describing historical surgical procedures or hyperacidity. The trap highlights that a vagotomy, which involves cutting the vagus nerve, was used historically to reduce acid production for managing severe peptic ulcers, demonstrating the direct reliance of parietal cells on vagal parasympathetic input. And to warn you about another trap, we're going to talk about peptic ulcer medication context. When we think about managing peptic ulcers or excess acid in modern clinical practice, surgeries are replaced by medications like omeprazole, which is a proton pump inhibitor that works by blocking the proton potassium ATPase pump. It is just a bonus trap for your information. Now moving to the exam pearl section, the cephalic phase functions as a highly specialized form of pre-absorptive conditioning, where the familiar phenomenon of salivation at thought of food is pure vagal reflex. This slide is your complete enzyme reference. and you should be able to fill it entirely from memory before you sit the IMAT. I want to walk you through it once more in full because the sequence matters as much as the individual facts. The story of digestion is told through this table. This table establishes a highly scannable master sequence mapping out each primary digestive enzyme alongside its specific site of action, target substrate, and final chemical products. The pathway initiates with salivary amylase operating within the mouth via saliva, breaking down starch to yield dextrins and maltose. Working in tandem with carbohydrate cleavage, lingual lipase acts as a minor input within the mouth, targeting triglycerides to produce fatty acids plus diglycerides. Now that the food bolus exits the oral cavity and transits downward, the digestive timeline shifts to the gastric chamber where protein processing begins. Within the stomach, the protease pepsin targets complex proteins, breaking them down into polypeptide chains. Once this partially digestive chyme is expelled from the stomach into the lower tract, the system relies on a massive influx of pancreatic tools to handle major macromolecular breakdown within the mixing chamber. Operating within the duodenum, pancreatic amylase breaks down starch and glycogen to yield maltose and dextrin. Simultaneously, the lipid processing mechanics are accelerated in the same duodenal zone via specialized pancreatic output. Pancreatic lipase acts within the duodenum to break down triglycerides, yielding exactly 2 monoglyceride plus 2 fatty acids. To ensure protein breakdown continues alongside fat processing, the pancreas discharges powerful duodenal proteases. Trypsin and chymotrypsin operate within the duodenum, targeting polypeptides to yield smaller peptide fragments. As these endopeptidases finish cutting internal bonds, a terminal duodenal enzyme steps in to clip the remaining loose ends. Carboxypeptidase operates within the duodenum, acting specifically on C-terminal residues to yield free amino acids. Now that these large polymers have been systematically packed into intermediate fragments within the duodenum, the chyme transit moves organically into the primary site of final breakdown and transport. The timeline transitions to the jejunum at the brush border, where maltase targets maltose to yield 2 glucose molecules. Right alongside this sugar gatekeeper, another localized disaccharide handles standard dietary sugars. Sucrase operates on the jejunal brush border, targeting sucrose to produce glucose plus fructose. To ensure all major disaccharides are successfully split at this membrane interface, a third specialized sugar catalyst is positioned in the matrix. Lactase acts on the jejunal brush border, breaking down lactose into glucose plus galactose for branched carbohydrate segments that bypassed all the upstream luminal amylases. The membrane anchors a highly specific structural cleaner. Isomaltase operates on the jejunal brush border just like before, targeting the complex alpha-1,6 bonds in dextrins to yield glucose. Finally, the remaining peptide fragments must be processed into their individual building blocks right at the membrane edge to complete nitrogenous breakdown. Aminopeptidase and dipeptidase act on the jejunal brush border, targeting peptides to yield free amino acids. Now, before talking about the example, pay critical attention to the high-yield exam traps I'm about to tell you. The first one, we'll talk about the lipase product confusion trap. You must remember the explicit product matrix trap. Pancreatic lipase, when works within the duodenum, yields exactly 2 monoglyceride plus 2 fatty acids. Then the carbohydrate monomer misconception can be a tricky one as well. Remember, amylases only yield intermediate dextrins and maltose. You must rely entirely on the downstream jejunal brush border enzymes to get true monosaccharide monomers. When comparing endopeptidases and exopeptidases, be careful not to confuse the specific products of these proteases. The trap lies in mixing up their targets. Trypsin and chymotrypsin cut internally to yield smaller peptides, whereas carboxypeptidase acts as an exopeptidase on C-terminal residues to yield true free amino acids. Now, as established in our exam pearls, if you're asked on IMAT, where is complete digestion of X achieved? The high-yield answer is almost always the jejunal brush border. Then let me remind you again about alpha 1,6 branch exception, because both salivary amylase and pancreatic amylase possess a narrow bond specificity that leaves branched connections untouched. The human body is completely dependent on the specialized inclusion of isomaltase at the jejunal brush border to split alpha 1,6 bonds. And that completes the information of this slide. Just as you need the enzyme side table, you need the absorption side table as well. For almost every nutrient, the IMAT will test you on exactly where it enters the body. For that, let me give you the logic layer before the table, because absorbing the logic means you can reconstruct the table even under exam pressure. The jejunum is the default absorptive organ. Everything that can be absorbed by a generic transporter gets absorbed in the jejunum. The only exceptions are molecules that require something special. That is, vitamin B12 requires the intrinsic factor, which is only in the terminal ileum. Bile salts require the ASBT transporter, which is only in the terminal ileum. Alcohol and aspirin are the only things absorbed in the stomach because of their unique lipid solubility in an acid environment. Water is absorbed everywhere, but the colon is where the final conversion from liquid to solid happens. Now let me walk you through the slide to have a sneak peek of every important fact. This slide organizes the full biological sequence of nutrient absorption, meticulously outlining each nutrient class, its specific anatomical absorption site, its transport mechanism, and its final physiological destination. The sequence documents that glucose and galactose are captured in the jejunum via the SGLT1 secondary active transport mechanism, routing directly from the portal vein to the liver. Fructose is absorbed within the jejunum utilizing GLUT5 facilitated diffusion, flowing similarly through the portal vein into the liver. Now that we have routed the primary simple sugars into the portal systems, the matrix shifts to nitrogenous monomers, which utilize an energy-dependent entry method. Within the jejunum, the free amino acids rely on sodium-dependent co-transporters, ensuring their systemic transport via portal vein to the liver. While these water-soluble building blocks enter the blood capillaries directly, hydrophobic lipid groups must be handled through a completely distinct structural pathway. Long-chain fatty acids plus monoglycerides are absorbed in the jejunum via simple diffusion, initiating a sequence where chylomicrons are formed to route the fats through lacteals into the lymph, terminating at the thoracic duct. However, the body processes shorter lipid carbon chains differently, allowing them to bypass this complex packaging route entirely. Short or medium chain fatty acids are absorbed across the jejunum or colon via simple diffusion, taking a functional shortcut through the portal vein directly to the liver. Because fat-soluble vitamins share the highly hydrophobic nature of long-chain lipids, they are forced to hitch a ride on this exact same structural pathway. Fat-soluble vitamins A, D, E, and K are absorbed in the jejunum via a micelle-dependent mechanism where chylomicrons are assembled to transit these nutrients through lacteals into the lymph. Conversely, the standard water-soluble vitamins are easily dissolved in the luminal fluid and follow a much more direct vascular path. Water-soluble vitamins are absorbed mostly in the jejunum through various mechanisms, incorporating some active pathways and some diffusion to route directly into the portal vein. As the moving chyme clears this massive upstream absorption hubs, the small intestine relies on its terminal boundary to execute the highly specialized recovery of complex microfibers. Vitamin B12 is absorbed exclusively within the terminal ileum via intrinsic factor mediated endocytosis, routing safely into the portal vein. Right alongside this specialized vitamin recovery gate, a critical recycling mechanism is anchored to conserve the body's metabolic detergent pools. Bile salts are reabsorbed in the terminal ileum via active transport driven by ASBT transporter, entering the portal vein to return to liver through the enterohepatic circulation. While vast majority of absorption is strictly restricted to these downstream intestinal segments, specific lipid-soluble compounds can cross the epithelial sheath much earlier. Alcohol and aspirin are uniquely captured early in the stomach via simple diffusion because they remain highly lipid soluble at low pH, draining immediately into portal vein. Finally, the enormous fluid volumes running through the length of the canal must be recovered by capitalizing on the solute gradients established by all these upstream systems. Water is dynamically recovered across the small intestine and colon. Small intestine or intestine absorbing 7 liters, whereas colon absorbing 1 to 2 liters. And they operate via osmosis as it follows established solute gradients to empty directly into the portal vein. That was quite a lot because it was the whole summary of this chapter. This trap is not a new one. We already have discussed it quite a few times, once in the stomach portion, then again in the absorption portion. Now again as exam trap in this absorption table. You must still remember that alcohol and aspirin represent the only significant nutrients absorbed in stomach, meaning all standard sugars, proteins and fats are completely excluded until they reach the lower intestinal tract. Another pearl I want to mention is about the fluid traffic paradox. While the large intestine is textually celebrated for its role in solidifying waste, the small intestine is the true workhorse of fluid balance, recovering a massive 7 liters of daily water traffic via passive osmosis compared to minor 1 liter processed by downstream by the colon. This is our final slide before we move to the high-yield IMAT-Style MCQ solving session. And for this slide, I'm going to fire through every trap in this lecture at speed. This is your consolidation pass. If any of this makes you pause, go back to the slide that covers it. They are all fair game on exam day. Your dedication to mastering these concepts perfectly prepares you for the test. Now let's solidify the entire matrix before our practice questions begin. As documented in our final summary matrix, salivary amylase breaks down starch specifically into dextrins plus maltose, acting on alpha-1,4 linkage only, and becomes completely inactivated in the stomach environment. The structural parameters of stomach absorption are restricted to almost nothing, establishing that alcohol plus aspirin function as the only distinct exceptions to this barrier rule. Now that we have marked the strict limits of gastric uptake, the summary flows organically into the critical cellular products synthesized inside the same mucosal wall. Intrinsic factor is produced exclusively by parietal cells, explicitly noting they are not chief cells, meaning any loss of this lineage leads directly to systemic B12 deficiency and downstream pernicious anemia. To ensure you can cleanly separate these mucosal components from accessory organ fluids, the narrative moves directly to the biliary system. Bile is characterized as not an enzyme, acting physically to emulsify fat, where a functional deficiency triggers a dual deficit of fat plus fat-soluble vitamin malabsorption. While bile manages physical emulsification, chemical downpackaging requires active pancreatic juices, leading to the strict rules governing enzyme secretion. The baseline parameter for Zymogens establishes that only proteases are secreted as Zymogens, while pancreatic amylase and lipase are secreted active. Now that we have separated active tools from proenzymes, the matrix maps out the chronological sequence required to switch on the protein splitting cascade. Pepsin activation follows a strict sequence where enteropeptidase on the brush border cleaves Pepsinogen to produce active trypsin, which then systematically activates all other Zymogens in the lumen. As these active enzymes reduce nutrients to their simplest units, the summary follows naturally into the specific membrane channels that capture monosaccharides at the epithelial border. Fructose uptake relies entirely on the GLUT5 transporter, explicitly noting it doesn't use SGLT1, requires no sodium co-transport, and operates via passive facilitated diffusion. While simple sugars utilize vascular channels, lipid products follow an entirely separate structural route based on carbon chain boundaries. Chylomicrons package long-chain fatty acids only to route them through internal lacteals, whereas short-chain fatty acids take a functional shortcut directly into the portal blood. As the remaining transit clears these upstream carbohydrate and fat absorption centers, the narrative routes us to the absolute terminal boundary of the small intestine for final salvage. The final capture of vitamin B12 plus bile salts is restricted entirely to the terminal ileum only, meaning any surgical resection of this exact segment results in both deficiencies simultaneously. Finally, the regulation of all these sequential, secretory, and motor events is consolidated under the master autonomic controller of the tract. The vagus nerve actively stimulates gastric secretion, showing it doesn't inhibit the process, which is clinically proven by the fact that a historical vagotomy results in direct acid reduction. And two examples to end this slide with. As established in our examples, the hormonal and neural triggers are perfectly matched to specific luminal contents. Secretin is driven by an acid stimulus. CCK is activated by fat protein stimulus, and gastrin is stimulated by a combination of protein plus distension plus vagal input. Then the cephalic phase functions as a specialized form of pre-absorptive conditioning, demonstrating that the entire digestive engine begins actively prepping for chemical processing before food even enters the mouth. It was an exhausting lecture, but I can guarantee you I have covered every single thing that might concern you in your upcoming exam. I don't want any stone left unturned because I believe so many of you guys are relying on my lecture solely for your preparation, so I can't keep any gaps. Even after that, if you have any suggestion, please feel free to mail me at tutor.mylinhq.com. Actually, tutor at. It was an exhausting lecture, but I can guarantee you I have covered every single thing that might concern you in your upcoming exam. I don't want any stone left unturned because I believe so many of you guys are relying on my lecture solely for your preparation, so I can't keep any gaps. Let us begin our high-yield question-solving session by analyzing the core enzymatic triggers of the upper small intestine. Question 1 reads, which enzyme is directly responsible for activating pepsinogen in the human duodenum? And the options are pepsin, chymotrypsin, enterokinase, secretin, and gastrin. The underlying theory rests on the protective Zymogen cascade of the exocrine pancreas. To prevent autodigestion of the pancreatic tissues, proteases are synthesized and packaged in entirely inactive forms. Once pepsinogen is secreted into the duodenum, it must encounter a specific membrane-bound activator anchored to microvilli of the intestinal brush border to safely spark the protein-splitting cascade. Therefore, the correct answer is C, enterokinase, which is an enteropeptidase. Let us evaluate why the alternative options are incorrect to solidify our understanding. Option A, pepsin, is an endopeptidase that operates exclusively within the highly acidic gastric lumen and is completely denatured by the time it reaches the duodenal environment. Option B, chymotrypsin, is incorrect because it is actually a downstream target of active trypsin, meaning it is a zymogen that requires trypsin to activate it rather than being the initiator of the cascade itself. Option B and option E, secretin and gastrin, are incorrect because they are peptide hormones responsible for signaling and coordinating fluid and acid secretions, not functional catalytic enzymes capable of cleaving zymogen bonds. Now that we have established how pancreatic enzymes are switched on inside the duodenal lumen, we can transition naturally to examine what happens when the vital secretions required for lipid breakdown are physically blocked from entering the mixing chamber. Question 2 states that a patient with obstruction of the common bile duct presents with fatty stools, that is, steatorrhea, and a prolonged prothrombin time. Which pair of consequences directly explains these findings? The clinical theory here links the anatomical patency of biliary tract to systemic lipid processing. Let's say this is liver, this is gallbladder. We have ducts coming from liver making common hepatic duct, and we have duct coming from the gallbladder, which is 60 duct. Then they merge and they create common bile duct. Okay. Whenever a physical obstruction blocks the common bile duct, amphipathic bile salts can no longer enter the duodenum to surround and physically break down large fat globules. This failure of emulsification severely restricts the available surface area for pancreatic lipase, causing uncleaved triglycerides to pass undigested into the colon, which results in steatorrhea. Furthermore, because hydrophobic lipids cannot be packaged into water-soluble micelles without bile salts, the absorption of fat-soluble vitamins is completely compromised. The acute loss of vitamin K absorption leaves hepatic clotting factors unactivated, clinically presenting as a prolonged prothrombin time. Therefore, the correct answer is B. Impaired fat emulsification and reduced vitamin K absorption. Evaluating the incorrect options protects our reasoning on multi-system questions. Option A is incorrect because pancreatic lipase and gastric pepsin are synthesized independently by pancreatic acinar cells and gastric chief cells, meaning a biliary plumbing obstruction leaves their production untouched. Option C is incorrect because hydrochloric acid and intrinsic factor are products of gastric parietal cells, which operate for upstream from the common bile duct. Option D is incorrect because glucose transport relies on a sodium-dependent co-transporter and vitamin B12 depends on gastric intrinsic factor, neither of which relies on biliary secretions for epithelial entry. Option E is incorrect because amylase is a carbohydrate cleaving enzyme whose secretion into the duodenum is independent of the bile duct pathway. Having resolved how macrolipids are physically emulsified and packaged before entering the enterocyte, we can step smoothly into the exact microscopic transport proteins on the apical membrane that handle water-soluble carbohydrate monomers. Question 3 reads, glucose and galactose share the same apical membrane transporter in enterocytes. Which of the following best describes this transporter and energy source it exploits? The options are GLUT5, GLUT2, SGLT1, PEPT1, SGLT1, and option A reads concentration gradient, B reads ATP hydrolysis, C reads sodium electrochemical gradient, D has proton gradient, and E reads direct hydrolysis of ATP. The underlying physiology dictates that while simple sugars are small, such as glucose and galactose, their highly polar hydroxyl groups prevent them from diffusing passively through the hydrophobic lipid core of the plasma membrane. To capture glucose and galactose against their steep intracellular concentration gradient, the apical brush border utilizes a secondary active transport. The apical brush border relies on a specialized co-transporter that couples the uphill movement of the sugars to the downhill electrochemical influx of sodium ions, which is continuously powered by the basolateral sodium pump expelling sodium toward the bloodstream. That means there will be one co-transporter over here, which is sodium glucose link transporter 1, and this is the apical membrane. In basolateral membrane, we will have sodium potassium ATPase. Therefore, the correct answer is C, SGLT1, sodium electrochemical gradient maintained by sodium potassium ATPase. Let us systematically dismantle the incorrect choices to reinforce membrane mechanics. Option A is incorrect because GLUT5 is highly specific channel dedicated exclusively to the facilitated diffusion of fructose, independent of sodium. Option B is incorrect because GLUT2 is a passive carrier-mediated transport protein localized to the basolateral membrane to handle the exit of sugars, and it doesn't utilize ATP hydrolysis. Option D is incorrect because PEPT1 is a specialized peptide transporter that imports intact di and tripeptides by utilizing a driving proton or hydrogen gradient, not simple sugars. Option E represents a classic exam trap. While SGLT1 is an active transport mechanism, it doesn't execute the direct hydrolysis of ATP itself, instead relying completely on the sodium gradient generated secondarily by the basolateral pump. Now that we have charted the active sodium-dependent mechanics that pull simple hexose sugar across the brush border, we can move organically downstream to evaluate the highly selective endocytic mechanisms required to salvage complex microfactors at the very end of the small intestine. Question 4 reads, where in the alimentary canal is vitamin B12 absorbed, and which substance is required for this absorption? The options are duodenum, and the enzyme name is salivary amylase. Option B is jejunum enzyme, or the substance bile salts, then terminal ileum intrinsic factor, stomach pepsin, large intestine mucin. The physiological theory highlights that vitamin B12 or cobalamin is a massive complex micronutrient that cannot utilize standard passive or active transmembrane channels. To survive the corrosive transit of the upper digestive tract, B12 must bind tightly to intrinsic factor, a protective glycoprotein secreted by the oxyntic cell or parietal cell of stomach. This intact complex travels completely unabsorbed through the duodenum and jejunum until it reaches the final segment of the small intestine, which is called terminal ileum, where enterocytes express highly specific surface receptors designed to recognize this complex via endocytosis. Therefore, the correct answer is C, terminal ileum intrinsic factor. Reviewing the incorrect options underscores the strict regional boundaries of the gut. Option A is incorrect because duodenum specializes in mixing and neutralization, and salivary amylase functions strictly to cleave internal alpha-1,4 carbohydrate bonds. Option B is incorrect because the jejunum functions as the primary absorption hub for standard micronutrients, and bile salts function to physically emulsify fats, not salvage cobalamin. Option D is incorrect because while the stomach synthesizes the required intrinsic factor, no significant nutritional absorption of the B12 complex can physically occur across the gastric mucosa. Option E is incorrect because the large intestine is functionally restricted to water recovery, electrolyte balancing, and microbial fermentation, completely lacking the receptor infrastructure for vitamin endocytosis. Having mapped out the structural endpoints where macronutrients and microfactors are systematically captured, we can transition naturally to the humoral signaling networks that detect incoming food to step regulate these secretomotor responses. Question 5 states, which hormone is released primarily in response to acid chyme entering the duodenum, and what is its principal digestive actions? The options are gastrin, cholecystokinin, secretin, motilin, and ghrelin. The underlying feedback logic relies on the fact that when the stomach completes its mechanical churning, it expels highly acidic chyme through the pyloric sphincter into the duodenum. Because the digestive enzymes of the small intestine are completely inactivated and denatured by low pH, the duodenum must instantly signal the pancreas to neutralize this incoming acid. This acidity is detected by specialized S cells in the duodenal mucosa, which immediately release a hormone into the circulation to stimulate the duct cells of the pancreas to discharge a massive volume of water rich in neutralizing bicarbonate ions. Therefore, the correct answer is C, secretin, and it stimulates pancreatic bicarbonate secretion. Let us evaluate the alternative choices to master the gastrointestinal endocrine matrix. Option A is incorrect because gastrin is released by G cells in the stomach antrum in response to protein and stomach distension, acting upstream to turn on acid production, which is the exact opposite of an acid neutralizing brake. Option B is incorrect because cholecystokinin is triggered primarily by fatty acids and amino acids entering the duodenum, focusing its action on gallbladder contraction and pancreatic acinar enzyme secretion rather than acid neutralization. Option D is incorrect because motilin is secreted during fasting states to drive migrating motor complexes, not in response to acute acidic chyme. Option E is incorrect because ghrelin is an orexigenic hormone released by the stomach during emptiness to signal hunger to the hypothalamus, and it doesn't coordinate duodenal neutralization. Now that we have seen how a localized drop in duodenal pH activates hormone-mediated pancreatic backup, we can progress smoothly to evaluate the systemic clinical collapses that occur when the terminal segments of this pathway are surgically resected. Question 6 reads, a patient who has had their terminal ileum surgically removed is most likely to develop deficiencies in which of the following? The options are vitamin C and folate, vitamin B12 and bile salts, glucose and amino acids, calcium and phosphate, vitamin A and vitamin D. The clinical theory demonstrates that the terminal ileum represents an absolute anatomical bottleneck for two highly specific physiological recycling and salvage pathways. It serves as the exclusive home for the surface receptors that internalize the intrinsic factor and vitamin B12 complex, and it expresses the active apical sodium-dependent bile acid transporters required to execute the enterohepatic circulation. If the segment is surgically removed, the body completely loses the ability to absorb B12 and can no longer recycle its bile salt pool, causing severe bile acid depletion that rapidly compromises fat emulsification in the duodenum, leading directly to fat malabsorption. Therefore, the correct answer is B, vitamin B12 and the bile salts, with consequent fat malabsorption. Dismantling the incorrect options highlights the functional division of liver across the intestinal length. Option A is incorrect because vitamin C and folate are water-soluble vitamins that are absorbed efficiently within the upstream segments of the jejunum. Option C is incorrect because glucose and amino acids are rapidly cleared in the upper tract, relying on the extensive surface area and tall villi of the jejunum hub. Option D is incorrect because calcium and phosphate absorption pathways are regulated by vitamin D and parathyroid hormone, primarily occurring in the duodenum and upper jejunum. Option E represents a classic exam trap. While fat malabsorption downstream will eventually hinder the uptake of fat-soluble vitamins A and D, the primary site of baseline micellar absorption is the jejunum, making their deficit a secondary consequence rather than a direct loss of ileal transport tissue. Having resolved the multi-system consequences that occur when bile salt recycling fails at the terminal boundary, the narrative flows organically back into the enterocyte cytoplasm to chart the metabolic fate of successfully absorbed long-chain lipids. Question 7 reads, which of the following correctly describes the fate of absorbed long-chain fatty acids in the enterocyte? The options are, they're transported directly into the portal capillaries as free fatty acids. B, they diffuse passively across the basolateral membrane into the portal blood. C, they're re-esterified into triglycerides and packaged into chylomicrons that enter the lacteals. D, they're converted to glucose by gluconeogenesis within the enterocyte. E, they are phosphorylated and transported via GLUT2. The biochemical theory dictates that once hydrophobic long-chain fatty acids passively cross the apical lipid bilayer via simple diffusion, they cannot be directly discharged into the aqueous environment of the interstitial fluid or bloodstream without causing fat emboli. To safely transport the lipids, the enterocyte routes them to smooth endoplasmic reticulum, ER or SER, where they are re-esterified back into triglyceride. These neutral fats are then packaged alongside cholesterol, phospholipids, and the vital structure protein ApoB48 to assemble large lipoprotein particles called chylomicron, which exit the basolateral membrane via exocytosis to enter the white porous central lacteals of the lymphatic system. This is the lacteal. Therefore, the correct answer would be C. They are re-esterified into triglycerides and packaged into chylomicrons that enter the lacteal. Let's evaluate the incorrect paths to reinforce the carbon chain length rule. Option A and option B are incorrect because long-chain fatty acids containing more than 12 carbon are completely blocked from entering the portal blood capillaries due to their lack of water solubility and large package size. Only short and medium chain fatty acids take the direct portal shortcut. Option D is incorrect because human enterocytes lack the enzymatic machinery to convert fatty acids into glucose via gluconeogenesis. Option E represents a conceptual mismatch. Phosphorylation and glutotransporter mechanisms strictly reserved for carbohydrate processing and basolateral exhaust sugar export, having no interaction with lipid packaging. Now that we have traced how long-chain lipids are successfully packaged into chylomicrons to navigate the lymphatic system, we can pivot smoothly to evaluate the clinical and structural limitations that arise when a student confronts carbohydrate enzyme deficiencies on the microvilli border. Question 8 reads, which enzyme is absent or inactive in individuals with primary lactose intolerance? And where is this enzyme normally located? Here are the options. Before getting to the right answer, let's discuss the perspective a bit. The structural theory focuses on the prerequisite that all dietary disaccharides must be broken down into single monosaccharide monomers before any transport protein can move them across the enterocyte membrane. In the case of milk sugar lactose, this cleavage requires the specific action of the disaccharide lactase. This enzyme is structurally anchored directly to the microvilli that constitute the brush border of the jejunal enterocytes, ensuring that the production of glucose and galactose occurs right at the entry gateway of the AGLT1 cotransporters. Primary lactose intolerance presents a developmental or genetic downregulation of this specific brush border catalyst. Therefore, the correct answer is C, lactase, brush border of jejunal enterocytes. Let us verify the errors in the alternative selections to lock in our enzyme locations. Option A is incorrect because salivary amylase is an oral enzyme that initiates starch breakdown in the mouth, having no interaction with lactose bonds. Option B is incorrect because sucrase is a brush border enzyme specialized for splitting sucrose, and it is entirely absent from the gastric mucosa, which handles protein denaturation instead. Option D and option E are incorrect because pancreatic lipase handles triglyceride cleavage within the duodenal lumen, and pepsin is a gastric protease synthesized by chief cells, neither of which has any functional role or structural location matching carbohydrate monomer production. Having mastered the specific microvilli coordinates where disaccharides are packaged split into simple sugars, we can transition naturally to trace the exact vascular routing through which recycled detergents journey back to the liver. Question 9 starts with, what is the correct sequence of structures through which a bile salt travels from its site of reabsorption back to the liver? The mechanical theory of the enterohepatic circulation highlights the continuous conservation loop designed to spare the liver from the high metabolic cost of synthesizing brand new bile salts from cholesterol during every meal. As we established during our regional analysis, the active salvage of these amphipathic detergents is strictly restricted to terminal ileum. We meant to say bile salts are compared with detergents, and the absorption or salvage of these detergents is only done in the terminal ileum cells. Once the active ASBT transporter pulls the bile salts across the ileal epithelium, they come inside the enterocyte and they exit the basolateral membrane and immediately enter the local venous capillaries. These capillaries coalesce directly into the hepatic portal vein, establishing a direct high-volume vascular highway that carries the salts straight back to the hepatocyte for immediate resecretion. Therefore, the correct answer is B, terminal ileum, portal vein, and liver. Reviewing the structural pathways of the incorrect options prevents sequence confusion on admission questions. Option A is incorrect because it describes the lymphatic route, which is a structural pathway uniquely reserved for large chylomicron fats and fat-soluble vitamins. whereas bile salts are completely excluded from lacteose. Option C is incorrect because the jejunum handles macronutrient capture rather than bile salt recycling, and the hepatic artery carries oxygenated systemic blood from the heart to the liver, running in the opposite direction of the intestinal drainage. Option D is incorrect because the cecum does not possess the active transport machinery for bile salvage, and routing through the inferior vena cava would dump the salts into the right atrium of the heart, bypassing the required first pass liver processing. Option E is incorrect because the duodenum represents the initial site of the bile entry into the lumen, not its site of reabsorption, and returning fluids flow directly to the liver parenchyma before any subsequent restorage in the gallbladder. Now that we have tracked the complete circulatory loop that conserves our biochemical detergents from the ileum back to the portal vein, we can conclude our session by evaluating the precise chemical parameters that permanently deactivate the very first enzyme introduced during ingestion. Question 10 states, salivary amylase begins starch digestion in the mouth. In which region of the alimentary canal does this enzymatic activity definitely stop, and what is the reason? We have 5 options: esophagus, stomach, duodenum, jejunum, and large intestine. The physiological theory highlights that every enzyme operates within a highly restricted environmental window dictated by its tertiary protein structure. Salivary amylase, or thiolin, is adapted to function perfectly within the near-neutral environment of the oral cavity, where the pH ranges from approximately 6.8 to 7. As the swallowed bolus enters the gastric chamber, it encounters a massive influx of hydrochloric acid that drops the local environmental pH to a highly corrosive range of less than 3. This extreme acidity disrupts the ionic and hydrogen bonds, stabilizing the amylase enzyme, causing it to undergo immediate and irreversible denaturation, which permanently halts all oral carbohydrate cleavage. Therefore, the correct answer is B, stomach, due to low pH denaturation of amylase. Let us confirm why the remaining structural sites are incorrect to finish our high-yield lecture matrix. Option A is incorrect because the esophagus functions purely as a muscular conduit, where wave-like peristalsis exerts absolutely no chemical or mechanical denaturing pressure on moving enzymes. Option C is incorrect because pancreatic amylase enters the duodenum to actively resume starch digestion, meaning it complements the carbohydrate breakdown pathway rather than exerting competitive inhibition on a previously inactivated protein. Option D is incorrect because jejunal brush border disaccharides function downstream to split intact dimers like maltose and sucrose, having no protease activity designed to degrade luminal amylase molecules. Option E is incorrect because the enzyme is rendered entirely non-functional long before reaching the terminal segments of the large intestine. And after running marathon for a few hours, we finally complete lecture 11 on digestion and absorption. Every enzyme, every transporter, every hormone, every site, every trap is covered. Go back to your anchor slides, use this script to build the narrative in your memory, and when you sit down to a question on digestion, you will have the entire system mapped clearly in your mind. Good luck. And if you found the video helpful, please drop a like, comment, and obviously subscribe to support our channel. And if you want to have a complete preparation for IMAT, you can check the link in the description for all the study materials. See you in the next video. Peace.